Hello? Hello?
Hello, good morning.
Yeah. Good morning. Anju?
Yes,
Yeah. Okay. Can you hear me? Can you see me?
yes, we can see
All
your
good.
screen also.
Okay. Okay, all right. Yeah. So we will be starting soon?
Yeah, within 2 minutes we will start.
Okay, fine. All right.
Good morning, everyone. We shall begin with today's session. I, Munarisha Malik, once again take this privilege to welcome you all to the second day of National Bioengineering Conference 2020, organized by the Department of Biotechnology and Medical Engineering, NIT Rourkela. After successfully concluding session 1 and 2, we shall now begin with the third session of the conference. I would like to welcome Professor Bala Subramanian Sir as the chairperson for the current session. I welcome our first speaker of the day, Dr. Vinulal Nelson Sati, Assistant Professor, Amrita Center for Neuroscience and Molecular Medicine, Amrita Vishwavidyapatham, Kochi, Kerala, who is going to speak on human tissues and organoids in a dish, potentials and challenges in recapitulating the in vivo environment ex vivo. I hereby take privilege to introduce Dr. Vinulal Nelson Sati. As I have already mentioned, Dr. Vinulal Nelson Sati serves as an assistant professor at the Amrita Center for Neuroscience and Molecular Medicine, Amrita Vishwavidyapeetham, Kochi campus. He is a recipient of the prestigious Ramalinga Swamy Fellowship administered by the Department of Biotechnology, Government of India. He started his research career as a research assistant at the Institute of Biomedical Sciences, Academia Sinica, Taiwan, in the area of medical biotechnology. He obtained his PhD in bioengineering from Amrita Vishwavidyapeetham and was a visiting scholar at Nichols Lab at Rice University, USA. He did his postdoctoral training from Kelly's lab at the Center for Bioengineering, Trinity College, Dublin. After his stint as a postdoc, he joined as a faculty member at the Amrita Center for Nanoscience and Molecular Medicine in 2017. His research interest is in the area of regenerative medicine with specific emphasis on investigating the immunomodulatory and regenerative properties of adult stem cells, biomaterials, and their bioengineered constructs. Developing bioengineered niches for achieving self-renewal of hematopoietic stem cells ex vivo is another major focus of his research. He has over 30 peer-reviewed research publications and 2 book chapters to his credit. Now I request Dr. Vinulal Nelson Sati to enlighten the gathering with his knowledge. Over to you, sir.
Thank you, Monisha. Good morning to all. Thank you for that nice introduction. I'm starting here with an introductory slide which actually fascinated me to the world of bioengineering. This was an image, a confocal image, which I took during my PhD, which was nothing but the mesenchymal stem cells growing on a nanofibrous matrix. So, moving forward, one very important thing. We all have, whoever is attending this meeting, we all have one thing as a common among us. So the thing is, what is that one thing? Because as far as I understand, this is a bioengineering conference, and people who are attending this conference is from the bioengineering background or biotechnology background. So then, to answer that question, I'm very sure that we all wanted to contribute our best to the healthcare area, right? So, and as it is rightly shown in this cartoon, this year which gave us a lot of trouble in the form of infectious diseases. So, whether bioengineers can do something in the infectious disease area, of course we can. Yes. Yes, Nisha.
Are you changing the slides?
Yeah, you can't see that?
No,
Sorry
sir,
for that.
yeah, it's okay, sir.
You can't see it.
We can see the slides, but like we are still in the first slide, the introductory one.
All right. Let me just check quickly.
Not like a slide, sir. Just like a slides are there.
Slides are there, but it's not in a presentation mode. Is that right?
Yes,
what you
sir.
think?
Yes.
Exactly.
Yeah. Let me check. Can I just stop sharing and come back again? Yeah. Yeah. About now? Can we see that?
Yes, we can, sir.
The same as previous one, or?
Yes, sir. It's not in the
let me
slide.
check whether it's not in the slide. Okay. Let me see. Okay. Any change now? Can you see this?
Yes, sir. Yes, sir.
Yes. Yeah, and just confirm for me whether the slides are changing. Is it changing now? Yeah.
Yeah, they are.
Okay. So this is the slide which we just talked about, and you all agree, right? And we are, as bioengineers or biomedical technologists, we are trying to contribute ourselves the best to the healthcare area. And before I go into the depth, I just want to say a little bit about my area. We are the Kochi campus from Amrita Vishwavidyapita, and it's one of the first nanobio center with research and academic components, and with the motto of the earnest aspirant gains supreme wisdom, Shraddhavan Labde Jnanam. And the thrust areas of our center is, the first one is bioengineering and nanobiotechnology, and then nanotechnology and renewable energy, and then molecular medicine. In the bioengineering area, we started in the tissue engineering and stem cell research, and we have a very strong team working in the cancer diagnostics and therapy, nanomedicines, biomedical implants, nanotoxicology, and I'm sure that yesterday you heard the importance and the areas which we are touching upon, the biomedical implants using the nanotechnology. And in the nanotechnology and renewable energy area, we have photovoltaics, nanostructured thin films, etc. And in the molecular medicine area, basic and applied aspects of molecular medicine, molecular and cellular phenomena in biological systems, and disease prevention, etc. Okay. This background, this is how I outlined my today's presentation. So I would like to take you to the biomedical industry, not in the details of what exactly is happening, but in the aspect of cost, because I want to give you a why and how the bioengineering can make an impact, and we can contribute something into the healthcare. And second one is the phases in the development of a drug or a medical device. Again, that is not into the technical process and the cost involved in this process. And then we will discuss the tissue engineering world, and then importance of potentials of engineered tissues, potentials, and then the challenges, of course. And then I'll be concluding with certain conclusion and future remarks. I'll start with this slide, the biomedical industry, and I'm sure that is a confusing slide to most of you, and it's a lot of numbers there. And this is a fact which came in the New England Journal of Medicine, and it's a very famous journal, and you all might be familiar with this. So don't get irritated with this slide, because we just need to look into these numbers. The United States 119, and then when it comes to China it is 8.4, Japan 37.2, and of course India is there around 2. What is these numbers? These are in billions. Okay, the R&D expenditure by the public sector and the private industry in these countries. Our countries, I mean, this is actually the data is from 2007, 2012, so it would have increased to a significant amount, but I don't have that authentic information about it, so I decided to go with this one. So even if $119 billion US dollars, it's a lot of money, and even for a country like us, $2 billion is a lot of money. Okay, I just want to move to the next slide with that. It's a lot of money involved in the biomedical industry, in the biomedical research. And if you take our country, not all the states are doing great in producing certain things. I mean, the healthcare area, biomedical devices or drugs, but certain states are actively involved in, and we can see Gujarat, Maharashtra, Karnataka, Haryana, Andhra Pradesh, and Tamil Nadu, and my state, like the Kerala is not into the area right now, but I'm sure that in the coming years, most of our states will be actively contributing, actively involved in the biomedical industry. You might be wondering that there is no similarity in my title and the way which I'm talking now, but give me a couple more minutes, I'll come back to the area, but this is important. So the biomedical industry in India also, a lot of things are happening. With that, I'll move to the next slide again. So I'll start with a simple example. Take an active pharmaceutical ingredient. We generally call it as API, and the pharmaceutical companies will spend from 0.5 million to 1.5 million or more just for one active pharmaceutical ingredient development. That means that's just identifying a compound, just a compound, and doing its initial studies. 1.5 million US dollars. It's a lot of money. Now, to take the industry benchmark for a candidate success rate, after spending a lot of money and after after identifying that a lot of the molecules or the products which we come out is not going to work, we take a percentage of it to the phase 1 clinical trial. Okay, and see that figure, around 48 to 64% of the phase 1 will be a failure. Okay, and if at all you are passing that one, again in the second phase, phase 2, it will be 29 to 32% of that will fail there. And if you are fortunate enough and take your finding to the phase 3, and 60 to 67% of the findings are going to fail there. What is the reason? We are spending a lot of money in the initial phase, in the animal trials, and finally we are reaching into a clinical trial. Still, this is a failure rate. Of course, we can't avoid this failure because there is no direct mechanism where you can test an active pharmaceutical ingredient or a medical device in a human directly. So, and of course, you are going to apply this, whatever you identify or develop, in a human being. So that's the reason for why this much of a failure there. So one more quick slide here. You can see 2 peaks here. I mean, don't go into the details. The second peak actually talks about the clinical trial. And look at this figure. The study estimated that the median cost of bringing a new drug, and we know that we are familiar with n number of drugs, even we are taking a lot of drugs these days, and the cost of bringing a new drug to the market was 985 million, and average cost is around 1.3 billion. So if you take this 1.3 billion and split it into these 2 peaks, one peak, of course, when it is into the clinical trial, in the phase 1 to phase 3, it's really expensive. We can accept it. But at least a quarter of the whole money is being spent on the first peak, where the initial discovery, lead optimization, and some preclinical studies are happening. Alright? So there also a quarter, up to half of the money is being spent there. Okay. So I think at least you are now agreeing to the fact that we are spending a lot of money in the initial phase itself for the development of something for the healthcare industry. Okay, this is what I said about the peak. Now, there is something very interesting. Okay, look at the animal models and something which I am firstly, now I am introducing the organoid models. Organoids are nothing but in a dish, if you can recapitulate at least one function of a particular organ or a part of an organ, then we can call it as an organoid model. That is something which is currently coming up. Even still, many of us are working in the tissue phase. And if you are progressing, then we reach into an organ. It's not an organ, organoid. So, an engineered construct which can recapitulate some of the anatomical features as well as some of the functions. That's the organoid model. And the other one which we are all familiar is the animal model. And I mean, it's a lot of wording there. I'm not taking into the details of it. But one thing which I found very interesting is, see, even from the animal model, results often do not transferable to humans. That's the standard of experiments which we all are getting involved, but the results often not transferable to humans. So this was published in Science. So let's see now. So what is required? What we can do as a bioengineer? There need to be a paradigm shift from the 2D monolayer culture experiments to engineered tissues and organoids. Here in this slide you can see, in the left hand side, you can see monolayer culture systems. We all are working with this one, either in a petri dish or in a flask or in a 96 well plate, 12 well plate, 24 well plate. We do routine experiments on this. Even me, I am doing it. Okay, and on the other hand, you have animals. I'm fortunate or unfortunate enough to work on almost all these animal models. You have immunocompromised mice like nude mice and normal mice, and then you have rats, different species of rat. You have rabbits, and you have large animals like pig and goat, sheep, etc. And of course, in India, it's not very common to do dog as a large animal study. So many of us, after our initial discovery, after initial testing, most of us are forced to take it into animal studies and small animal studies, and then to a preclinically validated animal model study before we take certain experiments to the clinical trial, right? Now, if you can remember the peaks which the expense peak which I just showed a couple of slides back, quarter of nearly 1.2 billion is spent in that initial phase where you evaluate. What we are doing to evaluate our cell culture systems and maybe some of the systems like small animal models or a fish model. So there, and of course we are happy to spend this, but the problem comes when most of these are failing in the clinical trials. Then if it is going to fail, is there a different way of checking it with a low cost, or is there a way which can come out with a system which can recapitulate more like a human tissue conditions so that we can get less failure in the clinical trials? So this is a question for all the bioengineers. There comes the role of the engineered tissues. So let's see what is happening in the tissue engineering world. From the moment people understand, scientists understand the ability to generate human tissues in vitro from stem cell has raised enormous expectations among the biomedical research community, patients, and in the general public. When you talk about stem cells and all, the general public is so excited. You can try it out. Okay, so they're thinking that if you have the stem cells, you are going to make an organ and it can be replaced, it can be used to transplant it into a patient. But we haven't reached in that stage. And you have two beautiful images. One of them is a human brain organoid displaying complex morphology. This is an organoid, which is not my data, which was published in Science in 2019. So scientists are currently able to develop such complicated structures in a dish, which is similar to what you can see in an embryonic brain. And in my study, I was able to develop something which can recapitulate the engineered bone marrow where the actual hematopoiesis can happen. This is my area of study. So this is where the tissue engineering world is happening. And another side of this is People are trying to develop these tissues as replacement for damaged tissues. That's another different area. This we call generally regenerative medicine area. Now we are trying to see, can we engineer tissues which can recapitulate some of the in vivo functions of organs, tissues, or pathologies in vitro, so that we can use in an ex vivo condition, so that we can use these systems to evaluate our active pharmaceutical ingredient or medical device, so that for the cost which we spend, we will get a more reliable result. So now, why cells on a polypropylene dish? We know that it's a polypropylene dish. That's where we culture. Not okay for evaluating an active pharmaceutical ingredient or a medical device. This is my statement. I'm saying that this is not okay. We have to improve. I mean, people already started, but still, including me, still we have to stick with some of the assays which has to be tested in a traditional polypropylene dish, which is of multiple configurations now, 96, 365, etc., etc. Okay? If it is not okay, why still scientists use it? Okay, these are two major questions. I think you might also have certain arguments, certain disagreements, or answers to these questions. So I have some data to demonstrate why cells on a polypropylene dish is not okay for evaluating your prestigious, your precious active pharmaceutical ingredient or the device, medical device, which you develop by spending years and years of research. Okay, so this is some of the data which I want to share with you. So these cells, when you talk about cells, stem cells or any type of cells, they are very much sensible to the mechanical environment, their environment. Their environment includes the media conditions which you give, the matrix in which you grow, whether it's a plate or the oxygen conditions, the temperature, etc., etc. So to investigate whether the matrix is going to play a role in stem cell differentiation. Actually, we conducted, in collaboration with Trinity College, Dublin, Trinity Center for Bioengineering, we conducted a study which has not been published yet. What we did was, we were able to develop cells in two shapes in the same material. I'm not taking through the details of the procedure, but in the same matrix, you were able to develop two cell shapes, and the same material you were able to develop into matrix stiffnesses. Okay, and in the initial studies, you found that most of the cells, regardless of the softness or stiffness of the material, regardless of the shape of the cells, they are mostly viable. You can still see certain dead cells that cannot be avoided when we work in a bioengineered environment, but in the viability evaluation, there is not much of a difference. Now, see what is happening. When you seed mesenchymal stem cells on a soft hydrogel, which has the same composition, and the top row is how you see the cells. This is not on a polypropylene dish. The polypropylene dish is a highly stiff surface. Nowhere in your body there is an environment which is as stiff as what is there in a polypropylene dish. And nowhere in your body the cells are sitting as a monolayer on surface of a stiff surface. Remember this. The cells are sitting on a three-dimensional environment. So what is happening here? When you see it on a soft surface, you can see that this is a transcription factor, YAP, which actually generally in cytosolic, and it can work, it can sense, depending on the matrix stiffness, this particular transcription factor can go into the nucleus. And as demonstrated here, it's just been published by others as well, demonstrated here in the top row, you can see the last, look at the last image where you have the nucleus, the DAPI, most of the blue you can see, and most of your YAP is outside, but the same matrix, only the stiffness is different, and you put it on the surface, and the cells, you see the cell shape, the difference in the cell shape, the bottom row is showing how the cells is on a stiff hydrogel, and the top row is the soft hydrogel, how the cells are. And you can see, interestingly, all the YAP, the transcription factor, they moved into the nucleus. What it means? even though you give the same condition, the cells which you put into a particular environment is going to have certain behavior of the environment. How will you decouple? When you check the toxicity of a component, when you check the effectiveness of a component, this is important. Now, when you put, as I said, when it is inside the body, in our body, no cells are in a two-dimensional conformation. So now what we did, we tried to put all those cells which we got inside the gel, whatever we discussed now. And we, even inside the gel, we got the round cells in a soft hydrogel, round cells in a stiff hydrogel, spread cells in a soft hydrogel, and spread cells in a stiff hydrogel. We got these four conditions. And just now you saw when on top of a stiff hydrogel, the YAP totally translocated into the nucleus. But in any of these four conditions, the YAP is not moving. That means if you are looking at certain specific mechanisms, specific pathways, and when you evaluate it in a monolayer culture, you may not be getting the right result which you want. So there we need to move into bioengineered environments which can recapitulate something like more closely to we may not be able to exactly mimic the in vivo condition, but something more closer to the in vivo environment. Another example here, same you have round and spread, soft and stiff. You see, just to see what is the difference, in a soft hydrogel, all the cells, most of the cells try to, in a soft hydrogel, less number of cells turned into an alkaline phosphatase positive cells. That means in a stiff hydrogel, more than 30% of the cells, around 50% of cells inside the gel became alkaline phosphatase positive. It is just the cells are sensitive to its environment. In the spread, also it showed same kind of a pattern. That means I showed all these biomechanics related data just to say that, to answer my first question, why cells on a two-dimensional monolayer culture may not, on a polypropylene matrix, may not be the ideal mechanism or the ideal system for evaluating your pharmaceutical ingredient or the medical device. Again, here also you can see the cells actually showed a matrix related stiffness or a softness related expression change in some of the key genes. I'm not going to the detail. So SOST, FEX and CX43, which are in the osteoadipo, the transformation, the lineage differentiation, those genes comes there. But the take home message is they are playing a big role there. With that, now, before moving into the next type of tissue, I should say that my second question was, still, even though there are n number of accumulating evidence are there saying that this is not the best thing to do, why still scientists, including us, are still using this one? At the end of the presentation, I have that as the demerits or the challenges, but here also I want to mention, because that's an established system. The variations which is currently occurring when you try to develop an engineered tissue from lab to lab, from a batch to batch, is huge. Okay, as bioengineers, we have to work on this area to take something which can give us a off-the-shelf, and nothing is available off the shelf. If you want to do an experiment with cells and a dish, you have a dish available, throw some cells and do the experiment. Here, if you want to engineer, nothing is available off the shelf. You have to establish the system first. It is time and money costing. So now the bioengineering area has to improve and come out with something which can available off the shelf, so the scientists can just take it from just buy it and do the experiment. Okay, so with that, I am moving to another a type of tissue which we can develop in a dish. So the right hand side is the tissue which can be correlated with, which is highly mimicking the native ligament or tendon. I'm not taking you to through the details of what is unconjugated, what is conjugated. Basically, it's nanofibers made as microfibrous bundles, and the cells are working well. The cells are behaving nice on this one. When we do the immunostaining collagen type 3, you can see the collagen type 3 which is specific for ligament and tendon, and collagen type 1 is also there, and a lot of collagen is deposited there. And the point is, you can, if it is necessary, if somebody who is, because a lot of pathology is happening in the area of tendon, because tendinopathy, and there is a schematic showing here all type of pathologies from fiber crimping, apoptosis, adipocyte accumulation, changed PEG-GAG cytokines, fibrogenesis, hypervascularization, etc., etc. So much is happening. But is it possible to develop a tissue which can recapitulate some of these things? My answer would be yes, but we have to spend time and effort on that. So with that, this we published in back in 2017. Now we know that our aged population is not only aged population. Now even in the youngsters, you can see the osteoarthritis. Osteoarthritis is nothing but you have mineralization, damage in the knee joint, cartilage, articular cartilage, and a lot of people are struggling with this pathology. So this data shows that this is engineered bone in which the chondrocytes, that the cartilage turned into bone through endochondral ossification pathway. The long bones in our body is developed as bone through the endochondral ossification pathway. If you remember this, that means it will form a cartilage template and then slowly it will ossify. So when we talk about osteoarthritis, this is one of the major reasons. Think about the smooth articulating surface in your knee is getting mineralized. There comes the inflammation, pain, and cartilage damage. What is the reason? Now, if somebody has to study this, they have to go to an animal model. Is there something which bioengineers can do? Of course, we can recapitulate the developmental pathway like endochondral ossification, where the cartilage is turning into bone, and how a cartilage is turning into hypertrophic cartilage. Now also people are trying, but they are trying to study this on a petri dish. And those findings may not be translatable. When you test those findings, the molecules which you find as the best, when you take it in an animal study and when you take it in the clinical trial, it may not give you the exact the result which you expect. That's my point. So we can develop certain tissues, the tissues for osteoarthritis, as shown here. See it mineralized beautifully, and the micro-CT images are showing here, are shown here. So I'm not taking to the details of what exactly is done here, and that was also published in 2017. And now, a lot of issues are happening at interface. What is this interface? When you look at a joint, you can see a smooth transition from bone to a calcified cartilage to a cartilage and then a fibrous tissue. That's how your ligaments or tendon, ligaments are actually happening. From bone, and it's beautifully there is a transition, tissue transition from bone, which is a highly mineralized tissue, to a calcified cartilage, and then there is a cartilage which is the articulating surface, which is not at all calcified, and then you can see a totally fibrous tissue. That's your ligament or tendon, right? So can we do for those pathologies what is happening in the tendinitis, or the pathology is happening related to the joint, the interface tissues? Can we do something? Of course, we can engineer this tissue. This is a complicated tissue because not one type of tissue, where you have to engineer an osseous phase, a calcified phase, a non-calcified phase, and a fibrous tissue in one construct. But it can be developed. So if somebody want to study this one in a dish, they have no other option. They can do for only one type of cell, or they might try to do a co-culture of two different type of cells. So this is also possible. That's my point. Now, this study beautifully demonstrates you can engineer cartilage ex vivo. It may not be exact the same as that of a in vivo cartilage which you see in a pig or a rat or a human, but using human cells. Now, I have to bring one point here, because the diseases are, there was one time where there was generic drugs which can work in a population the same way. But now you know that a drug which is working on me may not be working in the same concentration and the same dosage in another patient, even though his or her symptoms are more or less the same. Now the medical field is moving to an area which is called personalized medicine, because we need to exactly know what is good for that particular patient. In that scenario, there is no point in going and checking your initial studies on an animal model, a small animal model like rat, because you are going to check something for a patient. And so there is a possibility that now you can get a biopsy of that patient, you can grow those cells, and you can test that your interesting drug or a medical device after making an engineered tissue. So here, so this is again, I want to bring in, just as I just explained the importance of the matrix stiffness here. You can see the difference in the presence of this particular chemical, this DMARC. This is nothing but a prolyl hydroxylase inhibitor, which can give you the similarish trend as that of a long oxygen condition. The point is, nowhere in, maybe in some of the lung cells, the cells are experiencing like that high oxygen tension. Most of our in vitro studies, we are throwing the cells in an incubator which is 20% oxygen, because the 5% oxygen, or an incubator which can have a control over the oxygen tension, is really expensive. See the difference? The first two rows are showing how the chondrogenesis is happening in 20% oxygen, regardless of the different concentration of the growth factors and the molecules, and below is the 5% oxygen. The blue, the intensity of the blue is exactly showing how good it is. That means how good the glycosaminoglycans are produced in the cartilage. See the oxygen tension itself. So even the 0% of DMARC, that is the particular chemical, you can see a difference from the top to the bottom. That means oxygen is playing a role. Are you going to recapitulate this in a two-dimensional cell culture platform? So we need engineered tissues. Now,
for bigot.
Now, hello? Hello? Can you hear me? All right? Hello?
Yes, we can hear you.
Yeah. So we have moved to, like, this image clearly shows the collagen type 2 staining of the engineered tissue, engineered cartilage. You see, we can get the collagen type 2 expressed in a dish. That means those who wanted to study something on cartilage, there is a tissue, but I'm not saying this is exact the same as a human cartilage, but what I'm trying to say is this would be better than a system where you are just culturing some chondrocytes and testing your drug or medical device. From there, now I'm taking you to one of the interesting area which I'm currently working is to recapitulate the bone marrow environment. Because you know that bone marrow transplantation is a life-saving procedure, and umbilical cord blood is a source of hematopoietic stem cells. But for the success of a bone marrow transplantation, the number of hematopoietic stem cells in the transplant should be in a specific quantity. That means in a particular quantity per kg, and unfortunately umbilical cord blood you won't be able to get that much. That means the amount of hematopoietic stem cells which you get from a cord blood will be good for an infant. There is no way you can use that precious sample in an adult which is waiting for a transplant. Now, what is the only possibility? There is a possibility if you can isolate these cells and expand. And there is a number of studies where they try to expand with different cocktails, different cytokines. Of course, they got many of the studies, they got an increase in the cell number, but after they transplant, they are not engraft. They are losing some of their function. The point which I am trying to make is, you can't just take cells which actually reside in your bone marrow in such a complicated, sophisticated, cytokine-rich environment and put it in a bath of media and expect the same kind of a result. So what I am trying to develop is, my hypothesis was I can get these cells expanded with functionality if you can engineer a niche which is similar to that of a bone marrow. So here some of the materials which I use. I have a nanotextile. Maybe some of you might have heard about this one, which was a technology, patent technology, which we have in our center. We use that one, that textile, and we have seeded the osteoblast on that because in the bone marrow you have mainly 3 types of cells there. The osteoblasts are there, mesenchymal stem cells are there, and the endothelial cells are there. Of course, the smooth muscle, vascular smooth muscle cells are also there, and you have osteoblasts. See that, and the bottom row is showing how it looks, and you can definitely see mesenchymal stem cells in this matrix, and see the bright field image and the live dead image and the scanning electron microscopy. If you are one among them who have looked into the bone marrow, you can see these stromal cells. See the in between? This is exactly the same as what you can see inside a bone marrow. And of course, you can grow the endothelial cells also in the same way in the engineered niches. I'm not saying it need to be in the nanotextile. It can be any matrix. And this is which we are trying to recapitulate the bone marrow like environment. So it's a larger image which is actually showing you how the matrix, the mesenchymal stem cells are giving a mesh network. When you see this, the hematopoietic stem cells into this, they are going to get attached. They are actually having a physical attachment with these stromal cells, and they are trying to, do their natural process. So here, now what we did is, in this engineered environment, we were able to isolate the cord blood derived HSCs, and we seeded onto these matrices, and we were able to see, this is a live dead saying, this is the same image which I showed in the beginning of my presentation, and you can see a difference in the cell behavior. That means we are getting one step more closer to develop an engineered niche which can recapitulate the bone marrow like environment. I'm not saying it is bone marrow, but compared to a 2D culture, or the other option which I have is an animal model, which has, because you can't try this without without destroying the immune system in that animal. So you have an immunocompromised small animal, or you have a 2D culture. Here we have an engineered platform where you have human cells there, and you can try it out. So see the difference here. One of the important things when you try to engineer a dish, and when you try to engineer a tissue, and when you try to have something in 2D culture, one of the important things which we observed is this particular thing, osteopondin. Osteopondin is a secreted molecule which can actually induce quiescence in the hematopoietic stem cells. So we were interested to see how much osteopondin will be released from these stromal cells in our system. You can see that in all the culture conditions. See the different media conditions I have given. You can see the white bar represents the 2D culture, and black bar is a 3D culture. There is a difference. See in the DMEM and in the chondrogenic condition, compared to a 2D culture, the cells in a 3D, this is normalized with the DNA. That means for a given amount of cells which is in a 2D is producing more osteopondin compared to the given same number of cells in a 3D. Why is it? You have given the same media. So one reason could be because of the difference in the matrix induced, matrix related signaling. Now, another interesting which we found is, you look at, this is how in the scanning electron microscope you can see in the engineered niche, when you see the hematopoietic stem cells, this is hematopoietic stem cells in a flask on your right hand side, and we have different media conditions, and you can see that number of cells in the first graph in different media conditions. You can see that is into 10 to the power 6 cells. You can see a dotted line here. You are getting an increase in the cell number, and they are viable as well. That's the bottom line. What's more important here is, what is interesting to you might be, and if you compare the expansion in a 2D flask versus a 3-dimensional engineered niche, is nearly 30-fold increase we got in an engineered niche, whereas less than 5-fold is what is we are able to obtain in a 2D environment. And with that, we are not only focusing only in these areas, we are trying to, one key area which you can work is in the pancreas, the cancer models. See, this is the cancer tissue which we got from the patient, and if you put it in the dish, you can see the cells. This is behaving like the metastatic. This is highly pancreatic cancer, which is highly metastatic. They are trying to show the the pattern of moving out to look for the metastatic site. So we have two systems. In the first system, of course, the cells are growing well and it is looking like a tumor. And in the second system, which is a hydrogel based system, engineered system, you can see, consider this is an engineered system where these cells, when you culture them inside this gel, they start coming out just like the native tissue. So if you have a therapeutic molecule against this cancer, I am very sure that when you test it in such an engineered system, will give you more reliable result, which you might be able to take it into a translational scale than comparing it in a two-dimensional platform. Now, with that, I have to tell that we are at infancy. Of course, we have produced a lot of, the bioengineers are helping in all ways to develop, alternatives to the damaged tissues, the engineered tissues for testing, but there are challenges. As I mentioned, lab-to-lab variations. If I develop a tissue, I might get a result which is not comparable with another lab which produced the same kind of a tissue. That has to be addressed seriously. And lack of off-the-shelf availability. We know that the amount of funding which we are getting these days, when you get with the limited amount of fund, you will be forced to test your hypothesis directly than spending time to engineer a tissue, which will take a lot of your time. And the expenses associated with the establishment of the engineered tissue is also huge. But that would come down once if we establish these tissues. Now, the major challenges are, of course, when you engineer a tissue, it is not having an immune system, it do not have an established vascularization, and it do not have a nervous system. This is a major drawback in the engineered tissues, but a lot of people, including our group, are working in the vascularization. The immune system is, we are trying to, that the scientists are trying to develop certain lymph nodes and thymus, etc., engineered tissues, but still that cannot be something that is not going to be something which is possible in the next several years. But these are challenges. And with that, these are my conclusions and future remarks. There is an urgent need for alternatives to monolayer cell culture system for evaluating drugs and medical devices. And bioengineered tissues and organoids are potential options in the place of monolayer culture of cells in tissue culture plastic. These systems may produce data that can be relevant to human body, and personalized tissue and organoid development is possible, and it is the future. Remember that more investigations are necessary to produce engineered systems that can recapitulate in vivo-like environment ex vivo. With that, I like to acknowledge my fundings, like my fellowship and my fundings, and of course, I thank all of you for your patient listening. Thank you very much. And I'm open to questions if you have any.
I request the attendees to put forward the question in the chat box, or you may unmute yourself and speak it up. Anybody? All right then. Thank you so much, sir, for such a wonderful presentation. It was indeed informative and had surely captured the attention of all the attendees.
Thank you very much. Yeah, thank you very much. All right. All the best.
Thank you so much, sir. Thank you. We'll move on to the next presentation. We have with us our next speaker of the day, Dr. Shrivastava, Associate Professor, Department of Pharmaceuticals, National Institute of Pharmaceutical Education and Research, Ahmedabad, to speak on bioengineered cell instructive collagen hydrogel patch for intervertebral disc repair and regeneration. We'll now take an insight into Dr. Shrivastava sir's journey so far. Dr. Shrivastava is a proficient biomaterials and tissue engineering researcher specializing in the development of biomaterial-based medical devices for tissue regeneration, cell separation, drug or gene delivery, and fabrication of in vitro disease models. Thanks to his technical expertise on macroporous cryogel technology, cell culture and separation methods, and collagen-based biomaterials, to name a few, he has established a niche of his own in the ensemble field of applied life. Speaking of his academic experience, He obtained his master's in biochemistry from the CSJM University, Kanpur in 2003, followed by PhD from IIT Kanpur in 2010, and a postdoctorate from the Department of Microbiology and Network of Excellence for Functional Biomaterials, National University of Ireland, Galway in 2013, succeeded by another postdoctorate from the Centre for Research in Medical Devices, National University of Ireland, Galway in 2016. He has been a passionate pedagogue, starting off as an adult lecturer in 2003 at the CSJM University, Kanpur, to become an assistant professor in the year 2016 to 18, leveling up to being an associate professor in 2019 at the National Institute for Pharmaceuticals Education and Research, Ahmedabad, Gujarat. He remains an active member of the research community with copious publications in reputed journals such as International Journal of Biological Macromolecules, Drug Discovery Today, Stem Cell Reviews and Reports, Acta Biomaterialia, Nature Protocols, and several more. He has also actively participated in quite a few international conferences and workshops worldwide, notably in India, Netherlands, Switzerland, Ireland, Finland, Italy, and UK, even clinching the Best Poster honors a couple of times. Unsurprisingly, Dr. Shrivastava has bagged numerous awards, grants, and honors throughout his career, including the prestigious Ramalinga Swamy Re-Entry Fellowship, DBT sponsored in 2017, Science Foundation Ireland Award for Research Work in Japan, IACA in 2015, SYIC IS EU Chair in Termis EU Conference in 2015, Young India Investigator YIM India selection for the meeting held at Pune in 2012, product patent for new cryogel device in 2010, among many others. Sticking true to his impeccable teaching and mentoring skills, he guided the student team that won the second prize at Able Best competitions sponsored by the Department of Biotechnology, Government of India in 2018. Unmasking creativity has always remained the motto of Dr. Shrivastava as he continues to direct his research to wider avenues and ride to the ever so tough challenges. Without any further delay, I'd like to invite Dr. Akshay Shrivastava to begin with the presentation. Over to you, sir.
Yeah. Thank you very much for your kind introduction, and I thank the organizing committee for inviting me in Bioengineering 2020. So thank you first of all for such a long introduction of mine. So today I'm going to talk about bioengineered cell instructive collagen hydrogel patch, what we are developing in our laboratory, particularly for the intervertebral disc repair and regeneration. I hope I'm audible, and so everybody can see the slides also.
Yes, sir.
Yes. Okay. So first, let's talk about the unmet medical need in this area, and probably many of us are suffering from the low back pain. And the major problem in the low back pain that it is caused because of the degeneration of intervertebral disc. The intervertebral disc is a cushion-like material between the two vertebrae in your spinal column. And the center part of the intervertebral disc is called as nucleus pulposus, and the surrounding region is called as annulus fibrosus. So there are two different type of tissues which are present in intervertebral disc. The centrally located nucleus pulposus is more like a hydrogel, and the cells living there are called as nucleus pulposus cells, which are having a chondrocytic nature. The region surrounding the nucleus pulposus is called as annulus fibrosus, and this region is more fibrous in nature, not hydrogels. not like a hydrogels. And the cells which are living in annulus fibrosis is called as annulus fibrosis cells, and they are having fibroblastic morphology. So having in the same organ, you see the two different type of tissues which are present. So in a healthy IBD, everything is fine, and the cells are happily living. There is no change in their morphology or in the physical shape of intervertebral disc. But because of aging or mechanical trauma, genetics of the individual, or probably the inflammatory reactions in the body, there are changes in the cellular function and the cell number, which leads to the inflammation in the intervertebral disc. And once the disc is inflamed, it releases several cytokines. And these cytokines, enhances the cascade of reactions, which ultimately leads to the bulging of a disc. And then finally, you can see in this GIF that this annulus fibrosis region tear, and the nucleus pulposus oozes out and push the nerves. And because of this pushing, not only because of the nucleus pulposus, but also the bulging of the disc, you see this bulge is actually pushing the nerve. And because of this, you feel pain. And it's a very, very horrible pain. Some of you probably may have experienced this. And this is actually the major problem. Now, how to solve this? So there are several treatment approaches are available, but if you talk about NSAIDs, these only gives the symptomatic relief, it reduces the pain, etc. The another clinical viable option which is available is the complete replacement of total intervertebral disc, and these are nothing but the metal implants, or the implants made up of from hard polymers, and they have their own limitations. The metal as an implant between the vertebra again causes a stress around the vertebra bone, and then the bone degeneration started. So it also restricts the movement of your spine column, because metal is not so spongy compared to the intervertebral disc. So the limitation is great, and that is something clinic currently need as a new material which can prevent either the intervertebral disc degeneration process or revert it by some means. The another option which is usually done at earlier stages is the initial tear when happens to the annulus fibrosis is the suture repair method. They do the suturing around the material. But there is one unique limitation with the intervertebral disc, that it has been seen in several reports that once you touch the intervertebral disc by some mean or do the mechanical injury, and suturing is also a kind of mechanical injury in the annulus fibrosis region. Once this happens, again it causes the inflammation, and because of that inflammation, the disc degeneration start at faster rate. So this is something very unique thing happening in the intervertebral disc. So again, there is a great challenge, and sutures are also not so good in that case. The another therapies, for example, gene therapy, cell therapy, people have been working, but none of these are actually in the clinics at the moment, and there are a lot of great potential in the cell therapy, particularly I would say, and some of these reports have shown in gene therapy as well. Particularly, these all are targeting the ECM producing genes, or those genes which can suppress the inflammation, etc. If you look at this tissue is rich in ECM proteins. And one unique thing I forgot to mention, that the nucleus pulposus is rich in collagen type 2. And the annulus fibrosis, which is the surrounding region, is rich in collagen type 1. Apart from that, they are also rich in glycosamine glycan, chondroitin sulfate, for example, hyaluronic acid. They are more they are present in the nucleus pulposus in larger quantities compared to the AF. So by cell therapy And the gene therapy, people are trying to enhance the ECM production to give the strength to that area so that they can withstand a lot of load in the spinal column, and then probably particularly repair the whole degeneration process. In the cell therapy, it's very, very is challenging as well as has a great potential because the cell number which is present in annulus fibrosis is very less. If I talk about real numbers, I would say it's around 4,000 cells per centimeter cube in nucleus pulposus and around 9,000 cells per centimeter cube of annulus fibrosis. So you see the cell number is extremely low. So that's why as soon as the inflammation occurs, it decreases the cell number and ultimately causes a disease. So by giving these cells which can behave or differentiate into an IVD type cells, probably that could be one of the ideal solutions. However, it's still not in clinics, and there are several reports out there claiming for the same, that it can do so. Another important area which I would say is actually a combinations of the cells, genes, and other material which are useful for making sutures or the intervertebral disc. Combined in a combined effect, there are new strategies coming up which can be clubbed in tissue engineering approaches. And if you talk about the tissue engineering approaches, they are mostly composed of the growth factors by changing the cellular functions. Nucleus pulposus or MSCs, people have tried just by giving bioactive scaffolds and matrices they have provided to stop this bulging and revert the degeneration process. In this, what they are actually doing, they are completing the demand of increased cell number and increasing their activity, so that the repair process could start by controlling the entire inflammation process and by enhancing the strength of the tissue. In this approach, you see that the cells, not alone probably with the therapeutic molecules or along with the biomaterials, could be injected or inserted in intervertebral disc, particularly AF region, to prevent the oozing out of nucleus pulposus. So the approach could be either the injection of cells or therapeutic molecules can be given in annulus fibrosis, so they can enhance the local functions of these molecules and start the repair process. Or you insert a tissue engineered material in the interior of the annulus fibrosis to prevent the further tear of the annulus fibrosis region. Once the tear has happened or is started, you can also, the newer approach is to provide a patch, so that patch made up of the biomaterials and probably the cells or some other molecules could prevent the oozing out of the nucleus pulposus, as well as it can also help in the repair and regeneration of that region. So it's like a healing of the wound in the annulus fibrosis. So with this, what we are doing here in our laboratory is trying to do the multidirectional approach to develop a kind of a patch which can prevent the oozing out of the nucleus pulposus and particularly suppress the IVD inflammation. And there is a great demand. It's a clinical viable option as well compared to the whole replacement of whole disc or on the suture method. Another thing, the challenge is that these paths should have the structural similarity and and have an anisotropy, follow the same anisotropy which is present in annulus fibrosis region. So for that, we are focusing more on developing the unidirectional alignment of collagen fibers. Another important component which we are considering is the hyaluronic acid, and it's a type of glycosamine glycan. I have already explained that this is also a major component in the IVD. Apart from that, the main rationale behind using the hyaluronic acid that it also suppresses the inflammation. And you may have read it in several reports, including cancer, where the hyaluronic acid role has been discussed in suppressing cancer. However, a careful choice of molecular weight is required. So this altogether combinations is more like a physical, giving a physical shape and bioactive nature to the material itself. Now, to further enhance the repair process, we are focused on, first of all, not isolating the AF cell alone, but looking for the unique cell type, and that unique cell type actually could help in the regeneration and repair process. And I will be discussing about that as well in our work, what we have done here. Now, the another ideal solution is the BMSCs. BMSCs has shown to differentiate towards the annulus fibrosis-like cells in several experiments. So we are also considering the usage of BMSCs along with the patch for the repair and regeneration process. Our first goal was to mimic annulus fibrosis features, and then we first looked at the rat tail and the IVD of rat and looked into the scanning electron microscope. You can see these are several aligned collagen fibers are moving. So to mimic these, there could be several solutions. For example, you can choose electrospinning, melt spinning, or extrusion methods for the development of the aligned fibers. And our choice of material was collagen. So why collagen? Because collagen type 1 particularly is present in annulus fibrosis. I have already explained earlier. So the annulus fibrosis is made up of collagen 1, and that's the reason we have chosen collagen type 1 for the fabrication of the patch. But if we use electrospinning, then what happens? Because of the temperature and the current, this collagen type 1 degenerate, and after degeneration, whatever fiber you get, it is actually a degenerated form of collagen type 1, which we call gelatin as well. So the gelatin content in the fiber is much more. And the similar report was published in 2005 saying that if you spin the collagen solution spin this under the influence of electric field, it actually forms gelatin, not the collagen. So the basic material integrity is not maintained by electrospinning, which is actually the state-of-the-art equipment. So to maintain the integrity of collagen type 1, we have chosen isoelectric focusing method. And what is that? That if you throw any protein molecule, for example, collagen solution here, it moves towards the anode or cathode, and it moves until they find their isoelectric point. And that each protein molecule, and collagen is a protein, so each protein molecule has a definite pH where they do not move either towards the electrode or either towards the anode or cathode, and that's what we call an isoelectric point, where the protein molecules are at a ziterionic front. By using this concept, we actually have developed a system where we can develop a long collagen fibers. Okay, so we have used collagen type 1. You can see this assembly. This is simply electrophoresis assembly, and then this is what we have made. In the center there is a glass slide. We have added 2 copper wires, and between two, so they act as electrode, and between these 2 copper wires we throw a collagen solution. Then after certain voltage and for an appropriate time, these collagen molecules arrange themselves towards their isoelectric point, and then at that isoelectric point we can pick up this long thread of collagen fiber. And then this collagen fiber was further modified with the hyaluronic acid of certain molecular weight by chemical cross-linking. So with this way, we have developed the collagen fibers. which will be useful for developing the patch of annulus fibrosis. Now, to justify that why these fibers are useful, made by the isoelectric focusing, we have done the morphological assessment by scanning electron microscope, and in the scanning electron microscope you can see that the collagen fibers are nicely aligned. They have very aligned structure. Even after the coating of HA, conjugation of HA at two different concentrations also, the alignment is maintained. However, we have observed the higher level of unidirectionality in collagen type 1 conjugated with HA at 1 mg per ml concentration. So we have done this analysis by using fast Fourier transform analysis of the images, and with this we have concluded that the isoelectric focusing is a good method to develop the aligned collagen fibers for the development of the patches. Now the next goal is to check whether is there any compromise on the alpha helical content of collagen, which usually decreases in electrospinning and becomes gelatin. So here you can see the blue line is a rat tail tendon. So we have taken that rat tail tendon as a control to show that they have very high alpha helical content at around 220 nanometer of wavelength. Now we have taken gelatin as a control. Now you can see gelatin, which is a purple line, and you see gelatin lost all the alpha helical nature of the collagen, and it's more like a beta sheets, and that's why the collagen degrades from alpha helical to the non-alpha helical, which is beta sheet form. But by using the isoelectric focusing, you can see that we have maintained the alpha helical content, and however, if by looking these, though there is not significant difference, but there is an enhancement in the formation of alpha helical, and more the alpha helical content in the scaffold will enable you give better strength as well. So both collagen 1 alone or collagen with HA have shown to maintain the alpha helical content in the collagen after isoelectric focusing. And same, the chemical nature of the presence of collagen and HA was also checked by FTIR analysis, and the signature peak were found to present, like for example, NH bond stretching amide 1 and amide 2 of collagen. There's a unique peak of COH. There are a lot of hydroxyls which are present in HA. You can see here. So with this, it is confirmed that developed scaffold, have a good morphological features as well as the chemical nature which is required for the development of the patches. Now the next challenge is that that this is intervertebral disc is a mechanically active tissue, and there is a lot of load, and it allows you to bend in several directions. Your spine can bend, and it's bent only because of the intervertebral disc. So for development of a patch also, we have to mimic the tensile property and stretching property of the annulus fibrosis region. As such, the outer part of the annulus fibrosis region have the tensile strength of around 3.9 megapascal, and the little interior it is around 8 or 9 megapascal. So with this single fiber, what we have developed, and we have done the analysis in wet as well as in dry condition, and what we have found that in wet samples you can see the collagen 1 alone is showing a very good tensile strength property compared to the collagen 1 HA and HA alone. If you go for the young modulus, also the collagen 1, it looks more spongy, and the reason is quite obvious because hyaluronic acid, it actually increases the sponginess in the material. And what we are actually looking for the development of patch, that the patch should have a very good spongy nature, so that any loads which develop on the IVD, it is bare by the patches, and patch does not come off. So patch is very elastic in nature, and you can see by adding the HA, we have made it very, very elastic. You see the same trend in dry samples. However, the strength of the dry patches are much higher than the wet, which is quite obvious. One can understand that in the dry condition, the strength what comes out is the real strength of the material itself. But once you make it wet with the aqueous buffer or water, you see when the water goes inside, it actually allows a lot of chain movement, and because of that, it is actually showing the less tensile strength in wet condition. Now, if you look at the IVD tissue and particularly AF, the AF is not like a nucleus pulposus where it is like a hydrogel and there is a lot of water content. In AF, the water content is very, very less. It's more like a fibrous, dried kind of a tissue, not very dry, I would say, but it's a humid kind of a tissue. And that's why we have done the tensile strength analysis in both these conditions. So we have concluded that the addition of HA increases the sponginess in the material, and then after making these kind of fibers, our next target was to isolate a unique cell type. And for that, here we are showing with goat lumbar IVD, but we have also conducted experiment on the rat as well as the mice IVD for the isolation of unique cell type. So we isolated the goat. This is the intervertebral disc. This is the end plates. In between it is the intervertebral disc. And then we have dissected the tissue right in the middle. Then you see these kind of concentric layers. And this is endless fibrosis. Center is nucleus pulposus. So we carefully cut the endless fibrosis region without touching the nucleus pulposus, because we do not want. We are actually focusing on endless fibrosis for the repair process. And once this tissue is out, we digest it using the enzymes. We grow them in the laboratory, and then we actually treat them with a mixture of growth factors, which can differentiate these into the unique cell types called endless fibrosis. And we compare the same, which is naturally present in the AF region. Then we seeded these on the aligned collagen fibers, and we look for the biological activity in the future slides. So the first goal was to isolation of AFPCs. Now you see these are the unstained. So this shows there is no false positives are present. This is isotype control. In the sorting of the cell, you see these many cells are live. So we have selected the live cells. We have conducted this experiment through flow cytometry, and then from these live cells, we have found that this particular cell, this is shown in dot plot, purple color dot plot. We call it a CD146 positive. Why CD146 positive we are sorting? Because there is another recent report published by Nakai et al. That there are unique cell type which are present having the tenocyte-like property, and tenocytes are the cells which are present in tendon. So our expectation is that if we can get a similar type of cells from AF region, then it probably will be better to use for the repair and regenerative process. And after a processing of the endless fibrosis cells and induction through your unique growth factor mixture, we have got a lot of CD146 positive cells in endless fibrosis. And then we have sorted it, and after sorting, we have done the cellular proliferation test, and we have also checked how these cells could align on collagen type 1 patch, which we have developed through isolated focusing. Now here you can see that the growth of cell is on a positive side, and it grows up to 72 hours. You can see they can grow cells up to 100% of the area what we have considered here. Now here on the right hand side, you can see the nice comparable images of aligned collagen type 1 having the cells, and you see the cells are nicely aligned. on the fibers. So this is, and on the left hand side you can see we have taken as a control that non-aligned form of collagen just by making hydrogels of collagen without isolectric focusing. You can see the collagen fibers are randomly oriented, and then it also influences the orientation of cells. So with this we confirm that AFPC cells have good proliferative capacity as well as they can align on the developed collagen type 1 patch. We further grown them up to 15 days, and we wanted to see what happens to the cell number both in the normoxic condition and hypoxic condition, because there are some other reports showing the hypoxia nature, hypoxia kind of condition which is present in IBD. Now, if there is no hypoxia, you see there is so many cells are growing, and so they are happily growing. So the proliferation capacity of these CD146 positive cells isolated from annulus fibrosis, which we are calling annulus fibrosis progenitor cells. So they have a good proliferative capacity, and they are also aligning somewhere because of the high cell number and growth. They are also, not following the exact cue from the alignment, and they are mixing with each other. Now, if you look at the hypoxic condition, which we have conducted at 2% oxygen, you see the cell number is controlled. And I have earlier told you that annulus fibrosis have only 9,000 cells per centimeter cube. So cell number is extremely low. So I think probably because of the hypoxic condition, the cell number is controlled in intervertebral disc tissue. And we have found the same. You can observe the difference in cell number as well as the cell number decreases, the alignment is also, you see here, is increasing on the developed patch. So our next goal to see what these AFPCs are actually doing on the fibers, and for that our aim to see if we can harvest the new properties from annulus fibrosis cells. And that's what we are calling them a unique cell type, which we have isolated from AFPCs, which we isolated from annulus fibrosis. So we looked for the tenocyte-like properties in the annulus fibrosis cells. So we look for teno modeling, we look for Shrivastava, we look for Mohawk. These are key signature proteins, the markers which are present in tenocytes. We also look for actin kind of protein, ACTA2, and what we see that at low concentration of HA, they are showing expressions in ACTA2 and SCX. However, high HA concentration actually suppressing these two particular markers. So we are further investigating why this is happening. But if you look at the teno modeling and Mohawk expression, which is a key expression of these cell types, you see the expression is coming out from all the scaffolds. And this data is normalized to the control cells as well as 18S. So this is a clear upregulation of these markers which demonstrate that the AFPCs, which are isolated from AF cells, having tenocytic properties, which is very good because the tenocytes also have ability to secrete the ECM protein in high concentration, as well as they adopt the fibroblastic nature, which is a demand of annulus fibrosis region as well. So we are actually engineering the annulus fibrosis cell to behave differently for the benefit of the repair process. And for the same, the material what we have developed is aligned collagen and HA hydrogel is also supporting the growth of these cells as well as maintaining their expression. So further, we wanted to prove what is the status of the developed scaffold, and for that we have done organotypic culture by using a rat intervertebral disc. So this is a normal intact IVD. You see this is a vertebral bone, the center white is the this IVD intervertebral disc. Now we have made this defect here. You can see the small hole what we have made, kind of a square cut we have made, and then we have treated with developed collagen and collagen HA patch to fill this square place, because this is the kind of a tear which what we are trying to mimic. And then we checked after attachment, we checked whether the developed material could help in could help in the mechanical integrity of the tissue. So this is a small video you can see on your left hand side, the top left. That is how we have conducted an experiment using ultimate tensile strength machine, and similarly all these samples were tested under the same load. So when after making a defect, what we have observed that the nucleus pulposus comes out from inside through this tear what we have made. So that confirms that this is how the nucleus pulposus comes out, and we have also observed the strength for the same. Now you see when the AF defect was created, you see the blue line here in the stress versus strain graph. So this is a small linear portion, and then it fails. In normal IVD, it has long straight line followed by the small plastic, and then it breaks. Similarly, after the treatment of collagen patch and collagen HA patches, we have shown that nucleus pulposus oozing out could be prevented, as well as what we have found that AF patch along with collagen patch along with HA at 1 mg is actually giving a much better response compared to all other patches. So this is also possible. I told you earlier that having HA here is actually benefiting in having the appropriate mechanical strength. So we can restore the mechanical properties of the injured IVD. And the same trend we have observed in the young modelers. We have also calculated the failure stress. I'm not going into detail of this, but the overall outcome from this experiment that the patch what we have developed with collagen and HA could actually restore the mechanical property, and the next challenge is to see if the patch could also maintain the cellular integrity or tissue integrity. For that, we have performed another experiment in organotypic. So we have again taken the rat IVD region, and we made the defect, you can see, and treated with the electrocompacted collagen 1 and GAG modified collagen patches. We did the overnight incubation for attaching the patch at the site of defect, and we have done histology for that purpose to see what is happening. So this is a normal IVD. You see here there's a nice AF region is present, but in the defect what we have created, you see the AF region is distorted. Now there is AF is broken from where the nucleus pulposus comes out from this region. Then we looked at the collagen content, and we found that the collagen is intact in the normal IVD, but here it is broken. All collagen sheets are broken, and then we have looked for the GAG content as well as its morphology in bifringence. Now look at what happens after the application of collagen 1 patch and collagen HA patch. You see in that collagen, you see now here in collagen 1 patch, this direct red staining is showing. Somehow this patch is trying to prevent and making a bridge between the gaps. Same we have observed in Elysian blue as well. We have got much better results both in collagen type 1 alone or collagen 1 HA used at 1 mg per ml concentration. Now with this way, we can say that the developed patches have capability to restore the ECM content of the AF tissue at the site of injury. We further looked at the HA component, how it is present, and this is a normal IVD. You can see some HA is already there. So and then in the defect it's created. So whatever patch we have developed, you see here in the collagen HA, these patches are actually having HA and trying to make a bridge of the broken tissue. So this is how we evaluated that what is the role of HA. There is a further role needs to be understood in terms of the functionality what we have proposing saying that HA could suppress the inflammation, and those in vivo study must before claiming that HA along with collagen type 1 could actually suppress the inflammation and enhance the repair process. So with this all background work, what we have done along with the collagen type 1 and collagen type 1 HA and the unique cell type, what we have found in terms of AFPCs, what we are calling having tenocyte-like properties. Now, if you look at the whole disc tissue, you will see that there are lamellas of annulus fibers. And each lamella is actually having certain angle to the other. And we have found that it is something like a 30-degree angle between each lamella. So we are developing patches of long threads of collagen, and then we are doing the cross-linking by using ammonia vapors, and we are making patterns with these hydrogels. We are making a parallel pattern. We are making patterns of these fibers in the 30-degree angle. Manually we are doing it, and you can see here that in 180 degree, which is parallel arrangement of the patches. Now this looks like a patch which can be applied in a disc tissue as thick as human or goat or large animals. Another patches you see they are arranged in 30-degree fashion to each other, and you see the parallel patches are giving 10 to 12 megapascal, quite high. However, the AF tissue tensile strength is around 9 megapascal. So both 180-degree arrangement as well as 30-degree arrangement between the fibers are giving the appropriate tensile strength to maintain the tissue integrity. So the next plan is for us is to have the behavior of evaluate the behavior of AFPCs, which is a unique cell type we have isolated on these developed patches, and we would like our next step is to evaluate its performance in large animal. See, these kind of patches and IVD is very difficult to evaluate in the small animal. So we are looking for the large animal facilities where we can do the preclinical studies. Particularly, I am interested in the sheep model. So that's it, and I just want to highlight what other projects we are doing in our group. We are in the area of spinal cord regeneration. A lot has been done, but our aim is to do faster axonal regeneration by again suppressing the inflammation and immunomodulation. So that work is going on, and recently we have published one paper on developing such kind of material which can show electroconductivity as well as immunomodulatory function. We are also exploring the role of hyaluronic acid on the breast cancer stem cells, and we are getting a very surprising results in terms of the role of hyaluronic acid in maintaining epithelial to mesenchymal transition and cancer stem cell function. We are trying to modulate by hyaluronic acid, and ultimately we are into the development of HA-based therapeutics as a bioactive. We are trying to harvest the bioactive nature of hyaluronic acid in cancer treatment post-surgery. The another couple of projects are we have started, we are very innovative, and we can say in terms of the new devices what we are developing. One is punctal plug for dry eye disease, and we have actually achieved a lot in this area where we have developed drug plug alone as well as with the drug. So this is also a drug releasing material what we have developed. Our students have won the best table award as a business plan, and we have also qualified for Big Birak technical round, and we are still in the process of getting the investors for developing these kind of devices. The another device what we have developed is an ultra sensitive electrochemical immunosensor. For that we have chosen KIM1 as a marker, and we have developed the first prototype of sensing these kind of markers, and it is as small as glucometer. So with this I would like to acknowledge my research group, my collaborations and funding. Primarily, Ankush, Heta, Navneet, Prakash, Bhavani, and Pranav have worked on IVD area. And I have started IVD area from my postdoctoral study under Professor Abha Pandit, and I'm still collaborating with him on several aspects of repair and regeneration of intervertebral disc, particularly endless fibrosis. I'm also in the healthy talks and collaborations with Professor Swarup at IIT Delhi, and I thanks to research funding generously from NIPER Ahmedabad, Department of Ministry, Pharmaceutical Ministry of Chemicals and Fertilizers. I have back two fundings from DST, and I am a DVD Ramalinga Swami Fellow, and I also thank the Center Instrumentation Facility at NIPER Ahmedabad. Thank you very much. Let me know if you have any questions.
We'll move on to the questionnaire round. I request the attendees to put forward the questions. So there's a question for you in the chat box. I'll read it out for you.
Yeah.
And the question is, in the mechanical study slide, the Young's modulus of dry patch with 0.5% HA had greater value than the patch having 1% HA. What could be the reason for this?
Let me go to the slides first. All right. Just a minute. Yeah. So, Young modulus we're talking about. Yeah. So, if you look at the 0.5 mg and 1 mg, there is not a significant difference in the Young modulus, both in the wet as well as in the dry form. So I actually, I'm sorry for that I didn't put the p values here. But the difference between HA at 1 and 0.5 mg is actually not giving the great difference in terms of the Young modulus here. So they are showing exactly the same.
Hope it answers the question.
I hope
Any further question? All right then. Thank you so much, sir, for serving us as a speaker for the conference today. Your willingness to share your time and expertise has greatly served to the success of this event. Thank you so much.
Thank you very much. Thank you, everyone.
We move on to the next presentation, and we have with us Dr. Sakti Swarup J to speak on ionic polymer metal composites as actuators and sensors for biomedical applications. Speaking about Dr. Sakti Swarup J, she is currently working as Senior Assistant Professor at the Center for Nanotechnology Research in Vellore Institute of Technology, Vellore. Before joining VIT, she was working as a postdoctoral researcher in the Department of Aerospace Engineering, Indian Institute of Science, Bengaluru, in energy harvesting and sensing capabilities of the smart materials for novel applications. She obtained her PhD degree in Aerospace Engineering Department and Chemical Engineering Department, Indian Institute of Science, Bengaluru. She received her MTech degree in Nanotechnology and BTech degree in Electrical and Electronics Engineering from Vellore Institute of Technology, India. Her PhD work is on the ionic polymer metal composite actuators for insect scale flapping wing micro air vehicle. Her current research interests are in smart materials in structure, biomimetic system design, micro and nano sensors, MEMS, devices modeling and fabrications, energy harvesting, unmanned aerial vehicles. Her academic interests are nanotechnology, MEMS, micro device fabrication technology, smart materials and structures, introduction to aerospace engineering, biomimetic design. I'll now pass on the mic to Dr. Sakti Swarup ma'am to begin with her presentation. Over to you, ma'am.
So, a very good morning to all of you, and thank you so much for inviting me to deliver a talk on Bioengineering 2020. So, I'm really honored to share some of my ideas with you. So, I'll start sharing the screen. So thank you once again for the nice introduction. So am I audible and my screen is visible?
Yes, doctor.
Yeah.
Please go.
Thank you. So today I would like to talk about a material called ionic polymer metal composites. So and how these materials are very interesting and how these can be extended to a variety of applications and especially in the area of biomedical field. So before starting my presentation, I have a gratitude list. I would like to thank my director, Professor Nirmala Grace from VIT Vellore, and all the faculty members who I have been working closely with in VIT, and also the work that I'm going to talk today, the material modeling and experiment analysis was mostly done in IISc Bangalore. So I have a big list of people who I have to thank, with starting from Professor Ranjan Ganguly to Girdhar Madras, Debashish Ghosh, Sujay Mukherjee, Nilanjan Chattaraj, and I also have my students Prabhat Swain and Junaid, who are currently working on the IPMC modeling and experimental analysis. So why do we need these IPMCs, or what is the need for these new approaches for biomedical applications? So one thing, when we look at some in vivo applications wherein it could be a minimal invasive surgery, or it could be a drug delivery system, or it could be a device that is externally giving some vibrations to the heart. So for any kind of a device like an actuator which does all this role, the most important aspect is that it should be able to maneuver, it should be able to navigate through the different organs in the body. So when you imagine that you have a material which is going to get inside your body, the material has to be very flexible. It cannot be very stiff. So that's one of the challenges that the traditional or the conventional materials have. And the second one is dexterity, which means that how well you do it with your hand. So you have to have a device or actuator or a sensor which could perform an action inside your body in the similar way how your hands would do. And of course the safety, stability, the size, it has to be very miniaturized, the cost, and how good you can scale the device, the scalability of the device. So all these are the current challenges of conventional materials or conventional devices that have been used for different biomedical applications inside the body in terms of actuators. And to make a very efficient and to make certain actuators work in real time, we need to go for new approaches, and that's the reason we also need new materials with certain requirements. So the first requirement is it should have a good compliance, which means that there are some certain safety protocols, there are certain rules, there are certain way in which you have to make the device, and so your material has to be aligned with those safety protocols. It has to be extremely lightweight, and to put it in a very simple words, it has to be a very easy to operate and easy to use instrument. So not much complication in terms of applying an operational voltage, in terms of seeing how much displacement it is going to generate, and also in terms of the signal being conditioned and processed, the feedback system attached to it. So all these things that are associated when you design an actuator for a micro gripper or for a catheter or for any other biomedical application. So all these complication has to be minimal, and above all, the most important thing is it should have properties which is similar to the human muscle and tissue. So it has to be biocompatible, and it has to also be generating certain behavior that human tissue or a muscle generally does. So that is the reason ionic polymer metal composites are a very attractive material for the biomedical applications. In fact, the other name for ionic polymer metal composite is artificial muscles, which means that you can use this and replicate motions of your actual muscle. So biomimetics or biomimicking a natural process is very apt for ionic polymer metal composites, also called as IPMC. So here we can see two proposed designs for IPMCs. So one is used for a minimal invasive surgery. So it's basically like actuator which is going to clamp an object. So you can also think it like a tweezer which is used to pick a certain object. So the requirements of this kind of a tweezer or this kind of actuator which does a very minimal invasive surgery is that it has to be very flexible, it has to be controlled in a very great manner, and it has to be biocompatible, it has to be lightweight. The electronics and the feedback mechanism that is associated with it should be very minimal, and all these are satisfied when you use IPMC. So the other example for IPMC is a catheter tube. So for an endoscopic surgery, when you use a catheter tube, so that can also be used. IPMC can be used for these catheter applications as well. So these are certain biomedical applications for which we can use IPMC. So what is these IPMCs? So IPMC is ionic polymer metal composite. So it has an ionic region in the middle, and there are two electrode regions sandwiching the Nafion region. So this ionic polymer, which is generally made by Nafion, is in the center, and then you have a metal electrode on the top, a metal electrode on the bottom, sandwiching the Nafion membrane. And in general, the metal electrode is platinum. So we can see the EDAX graph, which shows platinum as 73%. So this is a flapping of a low frequency IPMC actuator. So the flapping frequency is 5.2 Hertz, and this is purchased from Environmental Robots. The size of the IPMC is 7 centimeter length, 0.6 centimeter breadth, and 200 microns thickness. So if you look at the IPMCs and the different layers of IPMC, so we can divide IPMC as the middle layer, which is the polymer region, and then the electrode region, which is covering the Nafion region at both the ends. So the different layers of IPMC has a certain way of influencing the actuation performance. So if you look at how an IPMC works, so they have a fixed cations and a movable anion, fixed anions and mobile cations. So the fixed anions are present in the Nafion region at its backbone, and they are not moving, whereas the cations are moving. And in general, the Nafion region is hydrated. So the cations attract water molecules with it. So you can see that the cations form a cluster with water molecules. So when an voltage is applied, the cations move towards the cathode, and it pushes the water molecules attached to it while it is moving. So when the water molecules move along with the cations, this movement of water, the pressure that is created because of the movement of water from one side to the other side, is going to cause the bending. So this makes a point very clear that the actuation of IPMC is mainly dependent on the water content it has. So more the water content, more is the bending or more is the actuation. Less the water content, less is the actuation. So certain parameters of the IPMCs which determine the level of hydration or the level of water intake is important, because as I said, water uptake is the major parameter which is controlling the actuation. So the problems with IPMC, the disadvantage, I've been telling the advantages of IPMC. The main disadvantage of IPMC is if you're trying to operate it continuously in air, it will dehydrate. The water inside the nephron membrane will start dehydrating. So that is one challenge of IPMC. The second challenge is if it is continuously actuating, there will be different cracks that will form on the electrode region through which the water will get evaporated. So that's the second problem. And the third problem is if you go beyond a voltage of 1.23 volt, actuation voltage or the input voltage, electrolysis happens and the hydrolysis happens and the water is going to split as hydrogen and oxygen. So you cannot go beyond a certain voltage. So these are the major disadvantages of IPMC. So when we look at IPMC and say that the actuation is dependent on the water uptake, so how are we going to maintain a certain amount of water, or how well are we going to return the water inside the IPMC is a big question. And how do we ensure that there is no non-linearity, because as the dehydration starts happening, the actuation displacement is also going to change. So there will be a non-linear behavior. So how are we going to control it? So these are some of the major challenges of IPMCs that restricts its usage for real-time applications, though it has an enormous advantage. So in our work, what we tried to see was how well can we prevent the water from dehydrating, or how well we can return the water inside the nephron membrane. So that was one of the study that we did. And the second study that we did was on the different nanomaterials. So if you incorporate nanomaterials into the IPMCs, how are they going to have an effect? And also some more experiments on improving the actuation performances of IPMC, actuation as well as the sensing performance. So these are the main factors: the equivalent weight, the counter ion, the concentration of the counter ion, the pre-treatment procedures that we do for an nephron membrane, the degree of hydration, the temperature and humidity. All of these in fact affect the actuation and the sensing performance. This is the different parameters in the nephron membrane. And then if you look at the electrode region, again the type of the electrode, the dimension of the electrode, the material properties of the electrode material, also these also decide the actuation performance. So to start with, we started with the mathematical modeling of IPMC. So the mathematical modeling model was adopted from Rourkela and Leo 2007 model. So this is basically an energy-based method. So there will be it's based on the Hamilton principle. So we find the potential energy, the kinetic energy, the amount of work done, and then put it mathematically and come up with a second-order differential equation from which we can calculate the frequency of the actuator, the tip displacement of the actuator. So for our application wherein we are trying to use it like a micro tweezer or like a micro gripper, kept the cantilever in the IPMC in cantilever configuration. So one end of the IPMC is fixed, the other end is moving. So this is the Hamilton equation. Del T is the variation in kinetic energy, del V is the variation in the potential energy, and del W is the variation in work done by the external force. So I'm not going into the details of the model that we did, but what we did after the modeling was we tried to match whether whatever modeling we did is correct. So we tried to validate it with the Rourkela and Leo model, and we observed that it was exactly in match with the Rourkela and Leo model. So the first study after doing the structural modeling of IPMC is to understand the effect of nanomaterials on the electrode region. So why the effect of nanomaterials was an important study was, as I said, that there are two things that happen with IPMC. One is you cannot operate it at a very high frequency. So it is a very low frequency applications are the only thing that we can use IPMC for. In case we want to increase the frequency of the IPMC actuator, or in case you want to make the actuators work at a higher frequency without a structural damage, then you have to incorporate nanomaterials into it. So one objective was to increase the natural frequency, and second objective was if we apply nanomaterials on the top of the electrode material, the weight, the mass of the device, the total device goes down, but you get a better efficiency, as well as there is a uniform coating on the electrode region which will prevent the dehydration effects. So three IPMCs were compared. So one is the traditional IPMC wherein you have an Naphion membrane and which is coated with platinum, and the other two materials were ruthenium oxide Naphion composite and LBL CNC composite. So ruthenium oxide Naphion was done by the direct assembly process, and LBL CNC was done by self-assembly process. So different type, using different methods of depositing the electrodes, three different electrodes based IPMCs were simulated and compared with. So we can see that the platinum electrode, the Young's modulus is very low. In fact, it was around 30 megapascal, whereas with IPMC, with the incorporation of nanomaterials, the Young's modulus values shooted up. So with ruthenium oxide Naphion electrode, the Young's modulus was 300 megapascal, and with LBL CNC, the Young's modulus was 400 megapascal. So this increase in the Young's modulus obviously increases the stiffness of the membrane of the IPMC actuator, and this increase in the stiffness will affect on the frequency as well. So we get a higher frequency operation of IPMC. So we can look from this figure that the platinum IPMC, the resonant frequency is around 17 Hertz, whereas with ruthenium oxide and LBL CNC, it shifts to 45 Hertz. So you get a high range operation when you go for ruthenium oxide Naphion and LBL CNC. And also the deflection, if you see here, deflection is very high compared to that what deflection you get for an IPMC platinum at that particular frequency. So this gives an idea that you can incorporate nanomaterials and thereby increase the range of operation of IPMCs from a very low frequency to a very high frequency without compromise on the deflection. So this study, the summary is that when you go for a high frequency range, if you use nanomaterials, you can operate it without any structural damage. So the second study was the structural modeling of IPMCs, mainly to understand how good they can hold the water. So the holding of water is mainly dependent upon the type of cations, dependent upon the equivalent weight, the pre-treatment procedures, and the bonding between the sulfonic acid and the hydrogen. So all this is going to decide how much water is going to be inside the IPMC. So if you look at the dry IPMC, the dimensions are given here. So the length, width, and the thickness. The length is 29 mm, width is 4.49 mm, and thickness is 283 micron meter. But when you hydrate it, there is a volumetric expansion, and the length, width, and the thickness, everything is going to expand. So you can see that how the difference in the volume of the dry and the wet IPMC. So when the volume is changing, obviously the actuation parameters also change. So if you compare the dry and the wet IPMC, the deflection of wet IPMC is much higher compared to that of the dry IPMC, whereas the frequency is the other way around. So the deflection and frequency are always inversely proportional. So the frequency of the IPMCs, dry IPMC, is higher compared to that of the wet IPMC, and the mass of the wet IPMC, obviously because of the water intake, is higher than that of the dry IPMC. So looking at the effect of cations on the actuation parameters, we can see that the deflection of lithium cations is very much higher, and for cesium it is very low. So these lithium ions can hold more water molecules compared to that of cesium. So that is why their deflection is also much higher compared to that of the cesium. But any time when we make an actuator, we cannot just focus on the deflection, but we also have to look at the frequency. We also have to look at the actuation force by which the force that it's going to exert. So the actuation force will increase if only the material is very stiff. So always having a good displacement is not the only parameter that we have to look at. So when we look at this graph, can be seen that sodium, the Na, has relatively good deflection and the resonance frequency. So thereby you get a good frequency, you get a good actuation force, as well as a relatively good deflection. So to have a trade-off between the deflection and resonance frequency, sodium is the choice. And looking at the equivalent weight of Nafion, again, when the equivalent weight is very low for a Nafion membrane, it can hold more water. So basically, the Nafion membrane becomes very soft when it has less equivalent weight, and it becomes hard when the equivalent weight increases. So again, looking at this graph, to have a trade-off between 900 to 1100 is the best equivalent weight a Nafion can hold, so that you get a trade-off between the frequency and deflection. And looking at how the IPMCs are going to behave at a different water activity. So basically, IPMCs are going to have a temperature and humidity effect profound on them. So when the temperature is increasing, the bond between the IPMC will break and the IPMC becomes very soft. So the deflection will increase, the frequency will decrease. And similarly, when the water activity is increasing, then again the deflection is going to increase. So we can see that as the water activity is increasing, the deflection is increasing, and the deflection at 90 degrees and 30 degrees, if we compare, the deflection at 90 degrees is very much higher. It's almost 5 mm compared to that of 30 degrees, which is 2 mm. And similarly, as the temperature is increasing, the frequency is decreasing because the IPMCs are going to become more soft because of the viscoelastic nature the IPMCs has. So the frequency will obviously drop down. And the summary of this studies is that the type of cations, the equivalent weight, and the temperature influence the actuation parameters more than the design parameters. So the IPMC can be used for to use an IPMC extensively for actuator holding water. We can choose accordingly. So the best way is to choose sodium as the cation, to have the equivalent weight between 900 to 1100, and so this way you can make sure that the IPMCs can return much water. So the next study was certain experiments. So till now what we discussed was on the mathematical modeling of IPMCs, and then we moved on to the experimental analysis. So the IPMC is mounted here, and you have a mechanical shaker which is going to give a periodic load to the IPMCs, and based upon the load that is based upon the input load, the IPMCs will generate voltage. So this configuration will give you the sensing characteristics, and also the actuation characteristics is studied using the laser vibrometer here. So the IPMC sample is actuated with a certain voltage. In general, it is between 2 to 4 or 2 to 5 volt peak to peak, and when the IPMC is actuating, a laser beam hits on the top of the IPMC, and by how much light is getting reflected back from the IPMC in the cantilever configuration, we can predict, we can calculate the tip displacement and the frequency. So this is the setup, and the actuation studies was carried out using the laser Polytech PDV 100 vibrometer, and for sensing it is LDS magnetic mechanical shaker. So this is how IPMC will flap at in water. So it's around 2 Hertz. So this is whereas it in air, the frequency is around 16 Hertz. So same IPMC in water, it's going to have a very low frequency of around 2 Hertz, but in air it will have 16 Hertz. So we did analysis of IPMC in a dry condition, in wet condition, and putting it inside the water. So we can see that inside the water, the frequency of the IPMC is very low. It's around 2 Hertz. When you take it out from the water, and in the wet condition, the frequency is around 8 to 9 Hertz. And when you dry the IPMC and actuate it, the frequency goes up to 16 Hertz. So one interesting aspect of IPMC is IPMC can operate well in air as well as in water. And the advantage of it using in water is all these dehydration problems that we have been talking about is a major drawback when you're going to operate it in air. But when you're going to just put the same IPMC inside water, the dehydration problem is not an issue at all. So IPMC actually works better in aqueous environment, and we can see that as the voltage is increasing, the frequency remains constant, but the frequency is more for a dry IPMC compared to that of the wet IPMC. And looking at the deflection, you can see that the deflection of IPMC in the wet condition is more compared to that of the dry and whatever is there in the water. So 6 IPMCs were taken to test. So one with the minimum mass and is J1, and it also has the length, breadth and thickness. The thickness of the electrode is also minimum. It's around 12 microns compared to that of the other 5 configuration. So why this 6 IPMCs of different configurations were chosen is to understand how the metal electrode plays a significant role in terms of water uptake. So the thing that we understood was when the metal content is more. So we can see that the J2, J4 and J6 samples, the metal content is more. And when the metal content is more, it takes less water compared to that of J1 and J5, which has less metal content, and also it takes more time for the water to get into. When there is more metal content, so the water to get in becomes a little difficult. So it actually takes a more time for the water to go in. So these were the 6 samples, and we can see that J1 and J5, which has the less metal content, has the maximum water uptake compared to that of the J2, J3, J4 and J6, which has less water uptake. And the time it takes, so from 75 to 90 minutes, you can see that there's no much difference between 75 to 90 minutes for J1 and J5. But for the other samples which has a larger metal content compared to that of J1 and J5, it actually increased pretty much between 75 to 90 minutes. So this is an important observation which tells us that the amount of metal in the electrode region it has a direct relationship with the amount of water the IPMC can take. So once the IPMC is put in the water, it also curls. So it is not anymore stiff, it becomes very soft and also curls, and that again has to be considered. So IPMCs, once these IPMCs are kept in the cantilever configuration, the IPMC was tested for the how good it can actuate. So one important test that we did was we preloaded the beam. So before giving a voltage and actuating the beams, we placed some mass at the end of the IPMC actuator. Different masses were kept, and we tried to study which mass is optimum to get a better deflection. So what happens when end mass is loaded is a behavior called as fractionalization happens. So in general, cations and anions are separate, and the cations are going to move towards the cathode to get deflection. When the beam is preloaded or when the beam is pre-bent, the division of cations and anions becomes more profound. So the cations actually move much better towards the cathode due to the bending of the IPMC actuator. So that in a way enhanced the actuation. It also enhanced the deflection of the it enhanced the sensing behavior of the IPMCs. So here you have the 6 IPMCs, and we have the voltage that it generates. So the resonant frequency is here, and the voltage that it generates is here. So again, if you look at J1 and J5, which are the electrodes with less metal content, the potential is less compared to that of J2, J3, J5 and J6, which has more metal content and more voltage. So the effect of mass loading on the actuators. So as the load is increasing, the voltage is also increasing. So this is the voltage for the low frequency IPMC, and this is the voltage for the high frequency IPMC. You can see that the voltage is increasing as the load is increased, and the voltage of the mass loaded IPMC is around 0.88. So without mass loading, the voltage of IPMC was 0.5, and it increased to 0.8 because of the mass loading effect. But the frequency for different loads remains same. So the frequency values, if you look at it, they are constant, but the voltage value is increasing. So the next one we did was depositing gold on top of the IPMCs to understand how effect of nanomaterials affect the sensing behavior of IPMCs. So you can see that after the gold coating, one observation was the output voltage increased compared to that of the one without the gold coating. And second is the mass loaded. So we tried to load different masses. So one was a very low mass compared to that of the mass of the IPMC. So the mass of the IPMC was around 0.0780, and we loaded the same amount of mass at the end of the IPMC, and then we increased the mass, and then further we increased the mass. So up to one value, so 0.173 gram, the voltage increased, and after that mass, when further additional mass was included, the voltage decreased. So from 0.33 for a 0.0780 gram of end mass, it increased to 0.79 for 0.173 gram, but it decreased to 0.28 when the additional mass was kept. So this also gives an idea that pre-bending or the fractionalization is effective up to a point, after which it doesn't have an impact. So this also helped us to understand how much mass we have to keep and preload the IPMCs to get a better actuation and better sensing characteristic. So the summary of my talk is, so IPMCs, basically the behavior of IPMCs are influenced by the viscoelastic nature, by the properties of the polymer, by the properties of the electrode. So working on the properties of the material can enhance the actuation and sensing behavior of IPMCs. And if you if you want to increase the frequency of the IPMCs, that can be done by incorporating nanomaterials into it. And the water retention, which is a major problem in IPMC, can be controlled when we choose the right amount of cation, and the pre-treatment procedures and the equivalent weight of the material. And gold coating of IPMCs helped in increasing the conductivity of the IPMC compared to that of the uncoated ones. So with this, I would like to end my talk. So thank you once again.
Any questions from the audiences? You may unmute yourself and question directly. All right then. Thank you, ma'am, for such an enlightening and entertaining presentation on use of ionic polymer metal composites for biomedical applications. Thank you once again.
Thank you so much. It was an honor to be here and talk. Thank you.
Same on our side, ma'am. Moving on to the last presentation of this session, I'd like to introduce Dr. Alok Jain, who is going to speak on combined atomistic and coarse-grained simulation study of peptide-based nanostructures from atomistic details to a mechanistic interpretation. Dr. Alok Jain is an assistant professor with Ramalinga Swamy Fellowship in Department of Bioengineering, Birla Institute of Technology, Misra. He completed PhD in IIT Kanpur in the field of computational biology. He had done 2 postdoctoral research from the University of Constance, Germany, and from Max Planck Institute for Polymer Research, Mainz, Germany. Also, he worked in Indian Institute of Technology, Kanpur as a project scientist. His expertise in the field of structural bioinformatics, computational biophysics, computer-aided drug design, and biomaterials design. He received prestigious awards as Ramalinga Swamy Fellowship, Biophysical Society USA Travel Award. He published more than 30 research articles from the reputed journals and book chapters from the Royal Society of Chemistry, and had several conferences proceeding from national and international journals. I now request sir to begin with the presentation. Over to you, sir. Sir, you're not audible. Would you please?
Thank you very much, Monalisa, for such a elaborate and nice introduction. First, I would like to thank the organizers, Dr. Anju, to organizing such a fruitful meeting. It's really an honor to be part of this meeting. So, I am going to discuss. Okay, first my slides are visible.
Yes, sir.
Okay, thank you. Okay, so I'm going to discuss about the atomistic and coarse-grained study on pentapeptides, and I will discuss how atomistic and coarse-grained simulation can help us to understand nanostructural assembly. Yesterday we have two computational talks from Dr. Soil and Dr. Moijori. They discuss about the system biology and biomechanics based approach to understand different biological questions. I will discuss in different dimensions where we use computational techniques, but different computational techniques, and try to understand the detailed atomic and subatomic level. So I have to start with the peptide-based nanostructure. I don't think for this audience I have to discuss the importance of nanostructures. Peptide-based nanostructures are very versatile in nature. They can be arranged to various morphology, various shapes, like nanospiral, nanotubes, various fibrils, nanofibers, by varying various factors. The governing factors for peptide-based nanostructure are nature of side chains. We can play with various amino acids, which will affect its final morphology and properties. External factors like the temperature, pH, electric current, by which again we can modify their properties, and internal modification like introducing some functional groups. However, it's difficult to identify the effect of these modifications beforehand. So if we want to design some intelligent system, intelligent nanostructure, which should have specific morphology and specific surface property for some biological application, in most of the time we do go ahead with the hit and try approach. Modify these various parameters and check if we are getting the desired properties and morphology. But that approach is exhaustive, expensive, and resource consuming. And there is no guarantee we will be able to get the structure with the desired properties. So I will discuss one such study, how we can get the mechanistic and atomistic detail, how a peptide forms the nanostructure. And such approach can be employed to design other biomaterials. The peptide system which we have considered in this study was originated from the R1 peptide, which is a strong inhibitor of malaria phagocyte invasion. This R1 peptide, so we only consider the pentapeptide. It was the essential segment which bind with the apical membrane antigen. We also substituted and designed another peptide where central residue proline was replaced with the glycine. So as such, there was only one difference between these two peptides. In first peptide, which I am going to call proline containing peptide throughout the top, is having the proline, and the other peptide, that is glycine containing peptide, does not have any side chains. So this is the proline, this is the glycine. Now our experimental collaborator, Professor Sandeep Verma from IIT Kanpur, they did experimental study on these peptides. They observed proline containing peptide form nice spherical morphology, while for glycine containing peptide morphology was totally distal, or I would say the morphology was ill-defined. It was not exhibiting any specific morphology. They repeated these experiments by varying the polarity of the solvent from 90-10% methanol water composition to 50-50 methanol water composition. By varying the polarity, it does not affect the final outcome. Only now peptide started aggregating more strongly as we increase the polarity of the solvent. Then they approach us and ask how a single residue substitution can affect the morphology so drastically. This was a very interesting system because of the drastic characteristic of both the amino acid proline and glycine, and their effect on the final morphology. So we decided to take this system by a powerful computational technique called molecular dynamics and simulation. Before I will discuss about my result, let me briefly introduce about the molecular dynamics and simulation. This is a structure of aquabody which transport water molecule across the cell membrane. For the discovery of aquaporin, Peter Egle received the Nobel Prize in 2000. He discovered aquaporin in somewhere around 1992. Even in the early 20s, the exact mechanism how aquaporin transport the water molecule across the cell membrane was not very clear, because we were having the static crystal structure. And as we know, all the proteins are dynamic. We can understand their mechanism and their function if we study the dynamic nature of the proteins or enzymes. And now if we see the dynamic nature of this aquaporin, we can see if we concentrate on this yellow color water molecules, you will see how actually water molecule transport, what are the residues involved, what type of energy barrier observed by the water molecules. All these intensive detail were clear by the molecular dynamics and simulation. So that was just one example about the application of molecular dynamics and simulation. What is molecular dynamics simulation? It is a science to simulating the motion of a system, and where all the particles are interact with each other, and we study how they interact during the course of time. To explain molecular dynamics simulation, consider this a one particle which is having initial position and velocity R1 and V1. You can correlate it with the, just like playing the billiard or snooker. So we have various balls in snooker. Now, what we do in the snooker, we hit the ball by a stick. So in a way, we are applying some force on the ball. So once we apply some force, then it will attain new position and velocity. Using the Newtonian mechanics, which is as simple as F equal to ma, we can get the new position and velocity. As we know, acceleration is the first derivative of velocity and second derivative of the position. So if we integrate this equation by a small time interval delta t, we can get the velocity v2 and position r2. Now, this was the simplistic representation of a single atom. In the case of protein or any material science or any other system, we have thousands of the particles, and these particles having unique velocity and position. And all the atoms, because these atoms are connected to each other by various covalent interactions or non-covalent interactions, so they experience the force from its surrounding. So if you again go back to the example of snooker, if all the balls are connected by a spring, it is difficult to decide the impact of the force or direction of the force to which you should apply on the ball. So that's again the same situation we have it in the when we are dealing with such a large system. So in place of calculating the force, we calculate the energy, which is the negative gradient. The force is the negative gradient of the energy. So by which we get the information about the force. How we get the energy value? Where we use various empirical functions and the equations by which we calculate the potential energy, then we calculate the force, and by integrating Newtonian equations, we get the position and velocity at the later time t plus delta t. We repeat these steps multiple number of times. At last, we get collection of the velocity, position, energy. We call these things ensemble and trajectory, the time trajectory, by which we calculate various thermodynamic and kinetic properties. So that was just a brief summary about the molecular dynamics and simulation. Now I will be coming back to original system. Just to remind you, chlorine-containing peptide was formed as spherical morphology in the experimental setup, and glycine-containing peptide, the morphology was ill-defined. So we have started MD simulation at the atomistic level by arranging 125 peptide into 5 into 5 into 5 array in the XYZ direction. We simulated these system up to 1.2 microsecond. We also repeated the simulation by varying the methanol water composition to mimic the experimental condition. As such, our observation was also same like the experimental observation is being increased the polarity, and the peptide system start aggregating much more strongly. It was observed in both the system. So that validate our parameter, our models was able to reproduce the experimental observation. But that was not only our goal. We want to know why this substitution proline to glycine affect the morphology so drastically. We monitor the system very closely. We observe few characteristic conformation in the case of glycine-containing peptides. One of the characteristic conformation was presence of many band structure, and these band structure very well correlate with the number of intramolecular hydrogen bonds and end-to-end distance. As you can see, these band structure was observed. The number of band, we have the more number of hydrogen bonds, the end-to-end distance was close to 0.7 to 0.8 nanometer. That correlate with the presence of intramolecular hydrogen bond. And surprisingly, these intramolecular hydrogen bond observe only glycine-containing peptides as shown by the platter histogram. That explain why we observe band structure only in the case of glycine-containing peptide. Beside these characteristic conformation, we also observe several perfect dimer structure where two peptides form many interpeptide hydrogen bond from 5 to 7. And these number of intermolecular hydrogen bond again observed only in the glycine-containing peptides. So that lead to perfect dimer formation. Now, then it puzzles us what happened, how actually these band structure form perfect dimer structure. And again, what happens, why it is not formed the perfect fibril structure which should be formed by these peptides, because it is already start forming the perfect dimer structure. So we have analyzed end-to-end distance. This is one of the analysis we did where we calculate the distance between the terminal C alpha atom, residue first and residue fifth. And we perform this analysis for both the peptide system. For the proline-containing peptide, we observe one single peak close to 1.28 nanometer that dictate the extended conformation of proline-containing peptide. While in the case of glycine-containing peptide, we observe broad spectrum of distances. Some peptide were in the bent conformation, some peptide also exhibit extended conformation. Then we repeated this analysis for monomers, dimers, and hieromers. Which means If the peptide is not interacting with any other peptide, it means it is in monomer conformation. What was the distance distribution? If it is in dimer conformation, when two peptides interact with each other, what's the distance distribution? And higher one mean 3 or more peptides are interacting with each other. In the case of chlorine containing peptide, we have not observed any changes with respect to its aggregation conformation. While for the glycine containing peptide, we observe monomer prefer to remain in the band structure conformation close to 0.6 to 0.7 nanometer, while dimer and iomer exhibit extended conformation, the large end-to-end distance. So it means there is stretching upon aggregation. So these monomers have to stretch, and then only they can form the perfect dimer or iomer structure. And this phenomena observed only in the glycine containing peptides. Then we visualize many structure generated during the course of simulations. We observe there was many dimers were formed, but most of these dimers randomly arranged. Similarly, for we also observe many band structures were trapped between these dimer structure. Because of these two phenomena, it induces the randomness in the system. And based on this observation, we propose the model for glycine containing peptide. In the solution, they remain in the band conformation by forming the various intramolecular hydrogen bond, or some of the peptides also remain in the extended conformation. Then they form the dimer structure by breaking these intramolecular hydrogen bond and stretching from band to extended conformation. This is only possible if they get the enough energy to able to break intramolecular hydrogen bond. And we observe this phenomena during our course of simulation. That's why we observe many dimer structure. Then these dimers start interacting with each other, but they stuck into random orientation. They are not perfectly aligned. Or some of the monomer which skip stretching, so they not form the extended structure, they trapped between these dimer structure. So now from these unordered state to perfect ordered state, they have to break many interpeptide hydrogen bonds or intrapeptide hydrogen bonds, which require energy. Besides this, dimers, the movement of dimers is restricted by the slower diffusion. So that's why system is stuck into unordered state and it is not able to form the perfect fibrils or any ordered state. While in the case of chlorine containing peptide, we observe hydrophobic collapse, like we observe in the case of folding of the globular protein. Most of the hydrophobic side chain they comes close and they buried inside the core of this nanostructure. It can be evident by solvent accessible surface area of these hydrophobic side chains which continuously decrease. That suggests these side chains are now not available to interact with its surrounding, means solvent. That's why solvent accessible surface area continuously decreases. And possibly the spherical morphology is surface tension driven. So based on atomistic data, we able to explain two phenomena. First, because of the presence of many interpeptide hydrogen bonding and slower diffusion of dimers, the glycine containing peptide is stuck into another state, and possibly for chlorine containing peptide, it is because of the hydrophobic collapse and it is because of surface tension driven. But because of our limitation of atomistic simulation, that's about the system size and simulation time, we could not observe the spherical morphology like observed in the experimental setup. So we decided to approach this problem using the coarse-grained simulations. In the coarse-grained simulation, we scale up many different atoms and represent by big balls. So in place of 50-60 atoms for atomistic at the atomistic detail, now we have 12 to 15 big balls in the coarse-grained simulation. So by reducing the resolution, we can simulate larger system and for longer time. Like for atomistic simulation, we simulate 125 peptide up to 1.2 microsecond. Now in the coarse-grained simulation, we able to manage 1000 peptides up to 11 microsecond. And we have used the Martini 2.2 for speed for coarse-grained simulation. Now again, there was two approaches available. We can go with the ready-to-eat receipt, like we have to just like it is available in the market. We can have various food are available. We have to just heat up or boil in the water. We can get the eatable food. But does it taste like the if you prepare your food from the scratch? Because you don't have any control about its ingredients. The recipe is available and it's the fixed. Similar way, if there are available model and force fields were available, we can run the molecular dynamics in simulation, then analyze the data. But if it is able to do the same justice, it is able to reproduce the experimental or atomistic data. Sometimes it does, sometimes it didn't. We also stuck to the same problem. We tried the ready-to-eat model, like available model and settings. So there was two models were available in the coarse-grained setup, because we have to predefine the secondary structure of our peptide. We try to define by the coil secondary structure, or extended mean the beta sheet conformation. But both the conformation not able to reproduce the structure conformation or aggregation propensity. As you can see, this is the atomistic data. Protein containing peptide observe extended conformation, glycine containing peptide we observe the broad end-to-end distribution. They dictate its structure conformation. If we go with the coil secondary structure model, we observe only majorly the band conformation. There was no difference between the protein containing peptide and glycine containing peptide. Similarly, if we go with the extended conformation, again, there was no difference. The model is not able to differentiate between both the peptide. And also for the aggregation propensity, either it is not aggregating at all or it aggregating so strongly. So problem in this case, we have defined our structure. And this problem is not that significant if we are dealing with the like the protein, because most of the time the protein structure is well defined. It's unique structure, and there was some jingling of the side chains or some domain motion. So we won't observe such type of problem, but for a small peptide system, this problem is very prominent. So now we decided we develop our own model. So what we did, we calculated the pseudo torsion angle from our atomistic conformation. By taking the atoms of the peptide bonds, we define 5 pseudo atoms from the 5 peptide bonds. Then we calculate the torsion angle from the pseudoatom 1, 2, 3, 4 and 2, 3, 4, 5. So these two pseudo torsion angles we have calculated from our atomistic simulation. We can get the probability of these angles be fitted into a Boltzmann conformation, and we get the distribution and the potential energy. And then we fitted our potential energy into a cosine function, which we have implemented this mapped conformation in our cosine simulation. So what happens? This was the original distribution of the pseudo angle in the original cosine simulation. Now, once we made our atomistic conformation on the cosine simulation, it is able to reproduce the atomistic data for both the torsion angles. It was very well reproduced for the protein content peptide, and also for glycine content peptide is qualitatively able to reproduce the atomistic distribution. So that sort out our problem of the structure conformation. The second problem we had about the aggregation propensity. For that, we vary various non-covalent interaction by varying the bead types. You can just imagine if we change the bead type, we are changing its van der Waals interaction or electrostatic interaction. So we have tested many different bead types. We come up with this particular setup, which was able to reproduce the both atomistic and experimental observations. So in summary, in our model, atomistic data was used to reproduce the structure conformation. We made atomistic conformation. We tuned the non-volatile interaction by varying the various B and get the perfect aggregation propensity. The main advantage of our model was we can disentangle the individual contribution, which will be clear in my next slide. The major limitation of this model was there was no explicit hydrogen bonding. Again, that was also beneficial for us, and that will be discussed in the next slide. Now, if you still remember our atomistic observation, there was two factors we discussed, hydrogen bonding and dimerization, that inhibit glycine content peptide to form the order state. First, start with the protein content peptide. Now, we observe nucleation event close to 360 nanosecond, then it formed the dumbbell-like structure. We observe shape transition, it formed the spherical morphology, like observed in the experimental same micrograph. This shape transition can be explained by this gain and loss kinetics. So gain means some peptide from the solution can go back to the aggregate state. Loss means some of the peptide which were already formed in the this nanostructure, they are going back to the solution. If there is this dynamic nature, gain and loss kinetics, then only the shape transition is possible, which can be clear by this movie. You see that now these are small aggregates were formed. They start the nucleation event for this nanostructure formation. Then they start merging to each other and forming the one big aggregates. So we observe a dumbbell-like structure. But still the system is keep on evolving, keep on exchanging the peptides with the solution. So at last, there was shape transition from dumbbell-like structure to perfect spherical morphology. Now, if we will go back to our system of the glycine content peptide, remember this model does not have explicit hydrogen bonding. So, in the absence of hydrogen bonding, this peptide also formed the spherical morphology, and it was more dynamic than the proline-containing peptides that confirm presence of the hydrogen bond, but was one of the reasons why system stuck in the unordered state during the atomistic generation as we observed. The other observation we had about the formation of many dimers, and we proposed that it is reducing the diffusion of these peptides. Now, to check that hypothesis, in place of simulating the 1000 monomers, we simulated the 500 dimers. We artificially formed the dimers because this model does not have the explicit hydrogen bonding. It cannot form the hydrogen bond by itself. So we artificially formed these dimers and simulated the 500 dimers. So just if you compare these two structure and their dynamics, you see in this case when we have 500 dimers, it reduces its dynamic nature drastically. Hardly there was an exchange with the solvent. So if system is stuck to any specific conformation, it won't be able to repair. So it is just presence of the dimer. If we will have the explicit hydrogen bonding also, then system will be much more rigid and sticky. So there is no chance of the repair in the case of glycine-containing peptide. That's why that system was ill-defined in the experimental observation. So that so in the summary, what we will say, these two peptide systems, proline-containing peptide and the glycine-containing peptide, without explicit hydrogen bond, it's like a Lego. So rearrangement is possible. So we can have the various shapes or various design because we can, the system itself can heal. While the glycine-containing peptide with the 500 dimers, when they form the dimer structure, it's more like the Tetris. There is fixed and no rearrangement is possible. And any wrong move, your system will be stuck into a disordered state. So in summary of my talk, we observe the conformational transition is required, going from bend to extended conformation for glycine-containing peptide, and dimerization reduce the possibility of rearrangement, and for proline-containing peptide, we observe hydrophobic collapse and the surface morphology and the hydrophobic morphology, the spherical morphology is surface tension driven. With this, I would like to end my talk by acknowledging University of Constance, Max Planck Institute, and my collaborator, Christian Peter, Professor Sandeep Verma, and Christoph Globis, and funding agency and the program from the German DFG and Ramalingaswamy University. With this, I would like to thank you for organizer and your presence. Thank you. And I am open and willing to take any questions.
I request the attendees to put forward the questions. Anybody? All right then, sir. Thank you so much, sir, for such a wonderful presentation today. We're grateful for the time and effort you took to share your knowledge and experience with all of us here today. Thank you so much.
Thank you very much. It was an honor for me.
Same here, sir. We'll now move on to begin with the oral presentation. Before we begin, I request all the speakers of oral presentation to stick to the time limits of 10 minutes each presentation. I repeat, I request all the speakers of oral presentation to stick to the time limits of 10 minutes. For the first oral presentation, I would like to call Neha Kukreti. Ms. Neha Kukreti.
Hello, ma'am. Am I audible?
Yes, ma'am. Yeah.
Good afternoon, everyone. Myself, Neha Kupreti. I am PhD Research Scholar in Delhi Technological University, Department of Biotechnology. So, for my today's objective is a thermochemical pre-treatment study of stubble waste for value-added products. As we know that waste is generated everywhere in the world, and which includes different type of waste like industrial waste, agricultural waste, municipal waste, used oil waste, plastic waste, kitchen waste, food waste, and dairy waste. So, by the agricultural practices, we generate large amount of agricultural waste in the form of corn stalk, sugarcane stalk, nut shells, wheat straw, and rice straw. And agricultural sector is the largest generator of waste material. And in the agricultural waste, we have different type of animal waste, field waste, and agro-industrial waste. So, straw waste is the abundant agricultural waste product, and burning of stubble waste, which is setting up of the after the harvest, the raw material or the straw that remains in the field is burnt, and it releases large amount of harmful gases. Causes huge damage to human health. So there is a first picture which shows the field after the harvest has been done. Straw from the fields of Uttar Pradesh, and the first thing that I have done is the biomass composition analysis. Because first of all, when we start with, we have to do the composition analysis, because after that we have to follow the pre-treatment and detoxification, then after that fermentation, and we'll get the value-added product. As we know that the different species have different type of chemical composition, and it varies from each species of the plant. So the lignin fraction, that is the non-sugar type, is the material that holds together the cellulose, hemicellulose together. The methodology that I have followed is first that I have done the feedstock preparation, in which I have taken the material, that is the rice and the corn material, and then I have dried and grinded the material. Then it is used for the composition analysis, and I have grinded the material to increase the surface area so that it is easy for the analysis. Then followed, I have used NREL protocol, that is National Renewable Energy Laboratory Protocols, and AOAC protocol for the composition analysis. And in the pre-treatment, I have done the thermochemical pre-treatment method in the rice and the corn stalk to break down the lignin and the hemicellulose method. And then I have analyzed the sugar content by the DNS method with the use of dilute acid and alkali pre-treatment. So for the composition analysis, I have used NREL protocol for carrying out for the various other parameters, and for nitrogen determination, I have used micro-Geldal method 1883. It has three components. First is the digestion of the sample, then followed by the distillation, and then the titration. So in Geldal method, it uses the hot acid, after which the whole of the protein that is present in the sample is converted to ammonia, and then after the distillation process into the standardized acid, and in the acid we further titrate it back to give us the amount of the nitrogen that is present in the sample. For the ash, I have used the AOAC 2000 protocol, and these are the pictures of both the samples, the rice and the corn straw, before the ash content analysis and after the ash content analysis. Then I have done the thermochemical pre-treatment of both the samples, rice straw and the corn straw, using the alkali and acid pre-treatment. Now the results of the rice straw composition, we have the maximum amount of cellulose, that was 34.12%, hemicellulose it was 28.45%, and lignin 18.12%. In the corn straw, I have the maximum amount of cellulose, that was 36%, hemicellulose 26%, and lignin 19%. Now the data for the moisture analysis, it was 7.73% in rice straw, total biomass 92.26%, ash content 8.63%, nitrogen 0.9%, and protein 5.67%. So analysis is important at this stage, because further I will be using the making out the bioabsorbent at the later stage. And in the corn straw, it has moisture content that is 8.23%, total biomass 91.7%, ash 12.3%, nitrogen 2.10%, and protein 13.11%. Now, the analysis of after the thermochemical pretreatment, the sugar that I got from the alkali and the acidic pretreatment in rice straw, the maximum amount of sugar that I got was in 1.2% in alkali pretreatment condition. And in acidic pretreatment condition, I got the maximum amount of sugar in the 1%. So alkali pretreatment with 1.2% sodium hydroxide released the highest amount of sugar, whereas in acidic pretreatment 1% of in sulfuric acid released the highest amount of sugar in the rice straw condition. In the corn straw, sugar release with the help of the alkali pretreatment, I got the maximum 1.2%, and in acidic pretreatment condition in corn straw I got in 1%. So alkali pretreatment with 1.2% sodium hydroxide released the highest amount of sugars, whereas with acidic pretreatment 1% sulfuric acid released the highest amount of sugar in the corn straw. Now I will be doing the detoxification of the liquid hydrolyzate, and further it will be used in the fermentation process, and I will be making the bioenzyme. Now what I would like to conclude that several factors have to be taken into consideration while designing the pretreatment of the conditions of the biomass, and I have taken the dilute acid instead of the concentrated acid in the treatment, because if in the concentrated acid, it forms the largest amount of inhibiting compounds in the form of phenolic compounds, furfuryls, and it causes corrosion, and it has also have the high cost. In my treatment, I got the result with the alkali that is more effective than in comparison to the acidic treatment of the lignocellulosic biomass, and the performance with the alkali was better than with the acid. Thank you.
Any questions?
Hello, ma'am. Hello, ma'am. I would like to ask only one question to Neha.
Yes, sir.
Neha, while you were presenting your composition analysis, you were telling about total biomass percentage, right? What does that mean?
Total solids?
Total biomass percentage.
Total solids, sir, you are asking here, total biomass.
Yeah.
Sir, it is total solid.
Okay, fine. So out of that, you could able to recover almost say 92%.
Yes. After the removal of the moisture, I got the total solid, that is 92%.
Okay. Fine. Then you have been done both acid treatment as well as alkali treatment, right? Out of that, alkali treatment seems to be much better. Could you able to tell the reason why it is being like that?
Alkali treatment is better. I guess for the lignocellulosic biomass, alkali treatment gives the better result because the cellulose and the hemicellulose that is present in the biomass, it is disrupted in a better way from the lignin.
Fine. Which is not possible with the acid-based treatment.
Yes.
Okay. Thank you. Yeah.
Okay.
Any more questions? All right, we move on to the next oral presentation. I would like to invite Abhishek Indolia.
Yes, ma'am.
Yeah, please go on.
Is it visible?
Not yet. Yes, it is.
Okay. Good afternoon all. This is Abhishek Indolia, a PhD research scholar from the Department of Bioengineering, IIT Mesra, Ranchi, under the guidance of Dr. Raju Poddar. So here now I am presenting the oral presentation. The title is In Vivo Investigation of Superparamagnetic Iron Oxide Nanoparticles, Spines as a Theranostic Agent for Magnetomotive Optical Coherence Tomography. Here is the complete overview of the presentation, which includes the introduction, origin of problems, hypothesis, methods, results, conclusion, and the references. First of all, I will discuss about the MMOCT. Basically, it is a new type of optical imaging modality which performs high-resolution cross-sectional tomographic imaging of the internal microstructures and provides 2D as well as 3D images. Yes, it is a non-invasive, non-contact bioimaging technique based on the Michelson interferometry, and it can achieve up to 1 to 15 micrometer in situ and in real time. It is not new, but it is a functional modification of the conventional optical coherence tomography. It utilizes the magnetic nanoparticles. Yes, these are biocompatible nanoparticles and enhance the field of view and enhance the imaging contrast. So now move on to the next slide. This is the indigenous design of the MMOCT system developed at the BIT Mesra. Here in the right upper side, we can see the sample arm of the scanning stage, where the sample is to be mounted for the scanning, and the solenoid is there just near the scanning of the sample. This is the complete design. This is the complete design of the solenoid with its frequency generator. Solenoid is used for the external stimulation of the magnetic nanoparticles at the time of imaging the sample, and in our case, it is the spines. This is basically some problems why it is to be needed, because the biologically tissues imaging suffers a low contrast imaging between soft and hard tissues, and for require sometimes targeted and site-specific imaging. Low depth of penetration into the tissue is a major one. To overcome these problems, we make this hypothesis. In the era of multitasking, one nanoparticle plays a multiple role. So we synthesize multi-role contrast agent for the MMOCT. For this, super paramagnetic array of the nanoparticles spines is being synthesized. Here, this is the FDA-approved biocompatible magnetic nanoparticles, which is stimulated by the externally, that is the solenoid in our case, and also used as a contrast agent. Now comes to the methodology. First one is the synthesis of the spines. This is the schematic representation of the synthesis procedure of the spines, in which cathodic electrochemical deposition, this is a widely used method is being used. For these, all chemicals were reagents grade purity. It comprises in the two-step electrochemical and the chemical setup. After the possible reduction reactions and form the ferric oxide, then it will go to the chemical reaction to form the final substance, that is spine. For the characterization of the synthesized nanoparticles, there are several characterization techniques used for the knowing the actual size, their shape, phase, and the property of the synthesized nanoparticles. This is the MMOCT system. This is custom-built MMOCT system developed at the BIT Mesra. This is the schematic. There is a swept source laser 1060 nanometer wavelength, travels through the fiber to the coupler, which divides it into two, that is 50-50 ratio, and then further goes into the second coupler, which divides again into 50-50. Then the one part of the laser coherent light goes to the reference arm, that is there, and the second part of the light goes to the sample arm, where the sample is to be mounted. After that, it reflects back by the reference and the sample. Then it follows the same path and goes to the BPD, that is balanced photodetector, and this data is further digitally processed by the custom built algorithm and the system. There is a solenoid. Here it is. There is a solenoid connected by the frequency controller for the stimulating the magnetic nanoparticles into the sample by the magnetomotion. This is the 3D representation of that part of the system. Now move on the digital post-processing. After the data is recorded by the MMOCT system, the signal is to be digitally processed by the custom-built algorithm in which magnetic modulation occurs due to the solenoid. These causes the phase difference into the nanoparticles. After the Fourier transform, the MMOCT signal is filtered out in the real value from the complex one, and we get the final processed result of the MMOCT images. And imaging of the spines with SSOCT. This is the ex vivo imaging of the chicken breast tissue. You can see the effect of a spine on OCT image with respect to time. It is continuously increases. This image is in 0 minute. After then 10 minutes, you can see that it is continuously increased with the time to give the more information inside the tissue, and the corresponding A-line can confirm this. This should be represent the depth information of the tissue with respect to the time. You can see there is continuously increasing in the depth information to see here. Then MMOCT imaging with the spine. Now we move to the MMOCT imaging. This research work is done after getting the approval letter by the ethical committee of the institute. In this study, the aqueous spines are topically applied on the clean-shaped dorsal part of the species albinomyes. You can see that after scanning, the recorded data is digitally processed and the result is to be generated. This is the simple SSOCT images. From this, we can extract the MMOCT signal, in which only the magnetic particles is to be present there, that is the spines. So we can see with time the nanoparticles penetrate. You can see penetrate into the mice skin, and spines work as a contrast agent, which can easily monitored non-invasively in real time by the system. This is the time dynamic study. This is the diffusion study of the spines into the tissue with respect to the time. This is the control image. There is no signal found for the MMOCT image, and after the application of the spines into the tissue, the field of view continuously increasing with the respect to time in the tissue. The penetration graphs corresponding. This is the dynamic. This is the time dynamic study of the spines into the tissue. This is contrast to the noise ratio result. This result shows the effect of optimized modulated frequency of 30 Hz into the tissue. We can see that without excitation of the magnetic field by solenoid is to be minimum, and the magnetic excitation is to be higher than the previous one. And for the spine for target imaging by the MMOCT, this is the imaging of capillary phantom, which is filled by the solid agarose gel. After the processing, this is the MMOCT. In this, spines is not present there, and in this, spines is present. And this shows the shows the spines into it. This is the overlaid image of the spines, and this is the overlaid image of that and that. And this validates the frequency modulation of 30 Hz, which is we optimized earlier. And the right side, this corresponds the same but into the tissue, in which red color indicates the spine present into them.
Excuse me, sir.
Yes, ma'am.
You have 2 more minutes to go. Oh, fine.
Okay. So now I am concluded in this study. It is used as spines used as a theranostic, which enables a wide range of research and clinical applications. It is used as a contrast agent for enhancing the deep tissue imaging. In this, it is used as a therapeutic tool for targeted drug delivery and for the hyperthermia application. And in the biomedical applications, we can use one of the major advantages of the system and the spines. It can be implemented using a fiber optic component and integrated with a wide range of medical instruments, including catheters, endoscope, laparoscope, surgical probes. Also used for guidance of surgical interventional procedure, used where the conventional, excisional, and biopsy hazardous or the impossible. These are the references. Okay.
Any questions?
Yeah. So I have one question regarding the. Can you explain what is the role of frequency controller?
Yes,
that we are having in the system.
So when we optimize this frequency, there is a maximum magnetomotion occur into the nanoparticles. That is good for the contrast imaging for us, and to know the best image obtained from the deep tissue imaging by this 30 Hz modulation, frequency modulation.
So what kind of modulation are you using?
It is what kind of modulation? Sir, it is a frequency modulation which is externally modified by the solenoid.
Okay, fine. My second question I have, what happens to the contrast agent?
Sorry, in our case, this spines can used as a contrast agent, or it gives just a minute, sir. In our case, spines used as a contrast agent. We engineered this, and this continuously proves after the time diffusion that it continuously increases the contrast with the time, and gets the maximum penetration into the tissue with respect to time.
Okay, you said about diffusion. What, like, diffusion means, like, what kind of diffusion are you talking about?
So this is the diffusion of nanoparticles into the tissue, into the mice skin in our case.
Okay, yeah. Done.
Thank you so much. For the next presentation,
Thank
I would
you.
like to call Menon Divya Ramesh.
Am I audible?
Yes, ma'am.
Can you see my PPT?
Yeah.
Okay, thank you. Good afternoon, everybody. My name is Divya Menon, and today I will be presenting an in silico analysis of papillary thyroid cancer patient samples for HIF1 alpha responsive genes as a prognostic marker. So I am from Department of Biotechnology, Cochin University of Science and Technology, Kerala, and I have done this work along with research associate named Bindya, and my guide is Dr. Ajit Vengalur. So before I start off into my main work, I would like to give a small introduction about thyroid cancer. Thyroid cancer is a very common endocrine malignancy that affects the thyroid gland. It is the gland that produces the thyroid hormones, and the incidence of thyroid cancer is on the rise. And this disease is projected to become the 4th leading type of cancer across the globe. Now, there can be many reasons why thyroid cancer has been on the rise. It can be due to increased detection of the early tumors, the elevated presence of many individual risk factors like obesity, and also there is an increased exposure to environmental risk factors. Now, into thyroid is being divided. Thyroid cancer is being divided into follicular cell-derived thyroid cancer and the parafollicular C cell also there. So here I am focusing more on the follicular cell-derived thyroid cancer, which is of 4 main types, called the papillary thyroid cancer, follicular, poorly differentiated, and anaplastic. So here papillary and follicular thyroid cancer are called the differentiated form. They are the forms which are easily responded to radioactive iodine treatment and chemical treatment, and the poorly differentiated and anaplastic form are the aggressive forms of the thyroid cancer. But mostly papillary thyroid cancer forms 85% of all cancers, and its prognosis is very good as it is very much curable by the radioactive iodine ablation. But a small subset of patients tend to become resistant towards this treatment, and that happens when this differentiated form of the cancer, it proceeds towards a de-differentiated phenotype, and that's when this happens. So the reason behind this migration to an aggressive state, what is it, is the main question in my research. So I would also like to introduce the topic of hypoxia, which is my main part of my study. So hypoxia is a very important hallmark in cancers, because when a tumor, as it grows, the kind of oxygenation level inside the tumor becomes reduced, and there's a condition called hypoxia where the tissues are not oxygenated adequately. So what happens is that when a tissue or a cell reaches a state of hypoxia, it starts to adapt to the situation. It starts to regulate many processes which will help it to either receive oxygen or able to overcome the situation. So for that reason, when hypoxia comes into play, a very important transcription factor called the hypoxia-inducible transcription factor gets turned on. It is basically composed of 2 subunits, an alpha subunit and a beta subunit. Now the alpha subunit is very oxygen sensitive. So in the presence of oxygen, it gets degraded. It has a very short half-life of about a few seconds only. But when hypoxia is there, this subunit gets stabilized. It will bind to the beta subunit and it will move into the nucleus, and it will bind to the promoters of various downstream genes. So these genes will get turned on and it will help it to overcome this hypoxia. So it will regulate cell cycle regulation, anaerobic metabolism, pH maintenance, angiogenesis. So this is how it will help the cell to overcome hypoxia. So in tumors, it is known that hypoxia is a major hallmark, and HIPs are also very much present or expressed in thyroid cancer tissues. So keeping this in mind, I have wanted to analyze the association between this HIF1 alpha and its positively correlated genes with thyroid cancer prognosis. for which I have downloaded the data of papillary thyroid cancer patient samples from the cancer genome atlas. So in this, that cohort composed of 501 patient samples and all their clinical data, normalized expression data, etc. is available. And from this, I have used the attribute of HIF1 alpha as a search data set. I've used that attribute and have seen the various kinds of genes that are upregulated or downregulated in the presence of HIF. So as you can see, there's a volcano plot which shows more than 19,000 genes which are regulated. Using the Pearson's coefficient over here, Pearson's correlation coefficient, I have shown that many genes have been correlated with HIF1 alpha, positive as well as negatively. Here I have focused more on the positively correlated genes. And here is a heat map showing the top 50 positively correlated genes with the HIF1 alpha transcription factor. So keeping this in mind, I used a cutoff of 0.7. So all genes that had a positive correlation coefficient of 0.7 I selected. And that is top 25 genes. And I did another analysis is that I used the KEGG pathway analysis, which is also available in the Linktomics. In the Linktomics link interpreter tool, this tool is available in the link interpreter option of the Linktomics. And in that, it showed the major pathways that are regulated by these 25 genes. So that showed that I have displayed an image of all the pathways that have been upregulated, and here the enrichment ratio was from 6 to 30 approximately. So in this itself, some of the pathways that were upregulated because of these 25 genes were the thyroid hormone signaling pathway and major pathways in cancer. So this itself shows a correlation towards cancer development by the presence of these genes, and actually further downstream analysis will give a clearer picture as to which and all genes are involved in this. Next, after KEGG pathway, the gene ontology summary showed the different types of functions different types of genes that have been involved in this. It gives a detailed outlook of how each gene functions at the biological level, molecular function level, and the cellular component level. So the list contained 25 user IDs, and out of which 23 were unambiguously mapped to the 23 unique Hendrix IDs. And this Go summary was based on these 23 unique IDs, and accordingly, what are the downstream pathways or genes that were involved in all these major processes. Next, these 25 genes, I queried them in the C Bio portal of the TCGA. So the C Bio portal, you can query the mRNA sequence or the methylation status, anything. So here I queried the mRNA seq data of these 25 genes. That is, I wanted to come to know what is the expression of these 25 genes in the thyroid cancer database. That is especially the papillary thyroid cancer database. So what is the expression, the mRNA expression of these particular 25 genes in that database? So in that database, there were 482 PTC sample data, and out of which 166, that is 34%, showed an altered expression. So why I have given the term altered here is because some of these genes showed an upregulation, that is they were high mRNA, and some of them showed an low mRNA. So the term altered is because there are randomly, some were up, some were low. According to each sample, patient sample, some were up and so an altered expression. So altered expression, 166 of the patients showed an altered expression. This altered gene set patient sample showed a decrease in overall survival as well as decrease in disease-free status of the initial treatment. So here I have shown an image of the overall survival status as well as the disease-free status. So overall survival is that is the percentage of the people who are alive even after 5 years of the diagnosis of the disease. So here the blue line indicates no alteration in that gene set, but the red line indicates people who had shown an alteration in that mRNA, they had shown a decrease in overall survival. Similarly, disease-free is the length of time after primary treatment that a patient receives. So after they given the treatment, from the time of the treatment till 5 years, how much they are remaining disease-free. So here also they showed that the patients that had an alteration in this 25 gene mRNA, that had alteration in the mRNA in these 25 genes, showed a decrease in the disease-free survival. I have also cited one single example of AB1, where this AB1 gene, it is a part of these 25 genes. It is known to be prognostic in melanomas and breast cancer, but has not been reported yet in thyroid cancer. So it is showing a decrease in overall survival as there is disease-free, and hence this can be further analyzed to know if it is being prognostic in thyroid cancer too. Next I did is I checked up some clinical attributes in the same sample data set curated with these same 25 genes. I checked about the BRAF V600E mutation versus the RAS mutation, some cancer tumor stages, MESA score and sex ratio. Now the BRAF V600E mutation is very much correlated to PTC. That is nearly 45 to 50% of all PTCs has the BRAF V600E mutation. So I wanted to know whether this has a correlation with those 25 genes. And I found that the altered group had more BRAF-like status rather than the RAS-like. They had more BRAF V600E mutations rather than the RAS mutations. Then I checked about the cancer stages, and it also showed that the altered group, which is depicted in red in color, showed more advanced tumor stages as well as extra-thyroidal extensions. Of the percentage of samples, the altered group tended to show higher advanced stages of the cancer as well as more extra-thyroidal extension compared to the non-altered group. Next, I checked about the MESA score. Now, MESA score is a score which is used to detect or predict the mortality of the papillary thyroid cancer patients. So, it is distant metastasis, patient age. The full form of MESA is metastasis, age, resection, local invasion and tumor size. So, both these groups have got a MESA score, but here, even then, the altered group has got a slightly higher amount of MESA score compared to the non-altered group. So the altered group showed us indicating there was greater mortality in the patients. Next, I checked the sex ratio. Basically, in thyroid cancer also, females have a higher tendency to attain the disease, get the disease rather than males. And here also, the altered group tend to show that the females had a higher ratio compared to the males. So summary of my results are, I selected the genes that were positively correlated with HIF1 alpha, and it showed a differentially regulated pathways which may be prognostic in thyroid cancer. Patients with this differentially expressed gene set showed advanced tumor stages with extra thyroid extension, higher MESA score, higher percentage of patient sample with the BRAF V600E. Also, the curated set of genes were shown to be prognostic in other cancers. Like I had shown an example of AB1, ELK3 is also present in ovarian cancer, KPNA1, endometrial and so on, but not yet reported in thyroid cancer. Hence, a detailed analysis of this will help to evaluate the significance in thyroid cancer prognosis. My future work would also involve the epigenetic regulation of this curated gene set on the papillary thyroid cancer patient sample database. And I would also want to check the real-time expression of this curated gene set. It will be validated in the PTC cell line. I would like to thank my university for giving me this opportunity to do this work. And now I finish my presentation and it is open for questions.
Any questions?
So one question.
Yes, sir.
So can you give me some more details on the 25 genes that you have found it correlating with HIF1 alpha? Can you comment on the comparison among these 25 genes in terms of structure, in terms of genome sequence? Can you give some more information on this?
No, sir. Actually, I haven't gone into the details. I have just said from this heat map, as you can see, I have selected it from there. So I haven't gone into that. But since you have mentioned this, I will definitely keep this in mind and do it in my research, because I haven't gone into the genomic basis of each of these.
Okay. Followed with this, based on your analysis, what kind of speculation you can make out of the study that you have made, especially in terms of this the mechanism or possible outcomes of this study?
Even though directly I cannot say a single gene can be correlated, but if I could go into more details with the KEGG analysis, no sir, I think find out some correlation with any methylation enzymes and everything. Because actually a little more further study I did, and I did find out that all these genes are positively correlated with the DNA methyltransferase enzymes. So if I can find a correlation, because it is always known that in hypoxia, when hypoxia is present, those kind of tumors become resistant to radiation and chemotherapy. They become resistant. So when I see there is some methylation correlation also to this, I can actually check if that methylation is also involved. So even if I cannot find a direct correlation between a particular gene and my thyroid cancer genes, but this significant correlation itself will help a lot.
So any comment on gene switching on and off aspect?
Yeah, this methylation will help to do that, sir. If I could find out how methylation, so it is accordingly, with the help of epigenetic regulation, they are switching on and off.
Okay. Okay, thank you.
Thank you, sir. Okay, thank you for this opportunity.
Thank you, ma'am. We move on to the next presentation. I'd like to invite Aditya Dev Rajora. I repeat, Aditya Dev Rajora for the next presentation, please. Is Aditya Dev Rajora around? Aditya Dev Rajora? Alright, I'll call the next name, which is Divyashri Dinesh. Divyashri Dinesh for the next presentation.
Yes, ma'am, I'm present. Can I just have 2 minutes?
Is it
2 minutes, ma'am.
Divyashri Dinesh speaking?
Yes, ma'am, Divya Shridhar speaking.
Yes.
Just 2 minutes, ma'am. I'll just get ready for my presentation.
Okay. Divyashri Dinesh, could you please begin with the presentation soon? Okay, I'd like to call the next person, Apeksha Devadika. Apeksha Devadika?
Yes, ma'am, she's coming.
Since we are running in short of time, I request all the participants to please be prepared with the presentation.
Yes. Ma'am, we are starting in few seconds, please.
Yeah, your presentation is visible. You can.
You are not audible.
Yeah.
Please.
Am I audible now? Hello?
Yes, you're audible. Please go on.
Let's start the screen.
Yeah.
Is the screen visible now? Hello?
Yes.
Yes, it is. Yes, it is.
Okay. A very good afternoon to one and all present here. I am Anjana Ramohan from Siddhaganga Institute of Technology, and this is my co-presenter Apeksha from the same college. We are going to be presenting on microtubule disassembly in corneal endothelium subjected to oxidative stress, a hallmark of Fuchs dystrophy. These are the contents of our slides. So cornea is the clear window of the eye that transmits and focuses light, and endothelium is the innermost layer of the cornea and is vital for maintaining clear vision. This is where the FECD is being manifested. So FECD is a sporadic condition that can be inherited by genetic mutation, and it is characterized by alteration in cell morphology, progressive loss of endothelial cells, and accumulation of extracellular matrix called Gate and it causes corneal edema. Finally, all of these factors lead to blindness. Oxidative stress is known to worsen the case of FECD, and exact pathology of FECD is not yet known. Looking into the stages of the progression, in the first stage, it is atypically asymptomatic, and it can only be diagnosed through a random visit to an ophthalmologist. In the second stage, guttae are formed and the cell loss is gradual. In the third stage, there's painful rupture of blisters, and the corneal transparency is lost. In the fourth stage, the patient becomes completely blind, and there's decreasing pain. One of the most important thing is to know the difference between normal cornea and the FECD corneal endothelial cells. So the corneal endothelial cells are monolayer. They are in post-mitotic arrest, and wounding in corneal endothelial cells takes place by sliding and enlargement rather than actual mitosis. So you can see that in the early FECD, the cells are enlarged, and there are also guttae formed. In the late progression of the disease, you can see that all the guttae formed will be burst open, and then there's complete destruction in the endothelial layer, causing the cornea to become opaque and leading to blindness. One of the main important features of the corneal endothelium is the pump-leak mechanism through which it maintains the corneal transparency. The fluid is leaked inside the stroma by tight junctions, and it is pumped out by an active sodium and potassium pump from the stroma. In a dysfunctional cornea, due to the absence of tight junctions, there is a continuous leak of fluid inside the stroma, causing the stroma to be swollen, and this causes corneal opacity. But there is no pump of fluid outside back to the aqueous humor. This leads to corneal edema. All of this is maintained by PAMR. That is perijunctional actomyosin ring. It maintains the barrier integrity, and it is composed of tight junctions, adherent junctions, and desmosomes. Microtubule, as you can see in the figure, the red color structure they are the filaments that maintain the cellular cytoskeleton, and disassembly in these causes the contraction in the PAMR, leading to barrier dysfunction. This disassembly is caused by oxidative stress.
As microtubules are a part of cytoskeleton, they are essential for structural integrity. Their disassembly is known to induce actomyosin contraction in different cell types, and this occurs through upregulation of small GT phases called the Rho A. It is observed that Rho-specific GEFs are bound to the microtubules, and when these microtubules disassemble due to the action of oxidative stress, it leads to Rho A activation. Rho kinase is a downstream effector of Rho A, which inactivates MLCP by phosphorylation of a myosin binding subunit. MLCP is required to keep the phosphorylation of MLC in check, but when this becomes inactive, there is an unprecedented MLC phosphorylation. Increased MLC phosphorylation results in actomyosin contraction and increase in centripetal forces, which causes the disruption of apical junctional complex and disruption of the PMR, and the entire barrier integrity, which we explained in the previous slides, are completely lost. Due to this, these are the following hypotheses, keeping in mind that microtubule is very important for the structure of cornea endothelium, and it completely disrupts in FACD. So oxidative stress induces disassembly of microtubules in CECs and disrupts PMR, which causes a breakdown of barrier integrity, which causes corneal edema, and ultimately it gets to the state of blindness. Thus, identification of upstream and downstream signaling pathways of microtubule is necessary, and we need to get a clear understanding of the same on the PMR, which, if we meet these needs, we can represent and find a key approach for identifying molecular targets to overcome this barrier dysfunction in CE due to oxidative stress in FECD. To realize this hypothesis, we have the following objectives. That is, to dissect porcine cornea and isolate CECs by explant method, to characterize structure of PMR and ZO1 by immunostaining, and by this we can characterize CECs, to investigate the effects of oxidants and antioxidants on PECs via ICC studies. So the first protocol was the dissection of porcine cornea. The cornea was brought from slaughterhouse and it was dissected, and the corneal button was removed and corneal endothelium was isolated. And this corneal endothelium was further used as an explant for CEC culture.
In explant method of cell culture, we take the explant, that is the corneal endothelial monolayer, and it is taken in a flask under conditions that allow the cells to migrate and settle down on the culture medium. It will migrate and settle down on the culture medium, and then when the confluency has been reached, these cells were taken for ICC characterization. You can see that this is the 4th day and the cells have started to migrate. By the 12th day, the cells have been settled down and the confluency has been reached. So, characterizing of ZO1 and afactin. ZO1 and afactin are one of the major components of the PAMR, and ZO1 is a membrane-associated protein that seals the gap between cells and connects to the cytoskeleton. Hence, you can see the staining of ZO1 in the cell junctions, staining the cell borders. And afactin is a major component of the cellular cytoskeleton, and it is characterized by the circumferential bands located in the apical portion of cell-to-cell borders. Both of these are exclusive targets of corneal endothelial cells, and hence we can validate the origin of our PCECs.
The next images are microtubule structures. These are untreated cells. So this is to completely know the structure of microtubules. Here we can see that microtubules are in expanded form and they have the classic fibular structure. This can be clearly seen in 100x magnification. The next image was when we induced microtubule disassembly by treatment with H2O2 and an oxidant. So the microtubule structure was completely lost. The classic fibular structure is lost, and the cells can be seen with many spaces in between them. But these are the cells treated with NAC. NAC is an antioxidant and it scavenges superoxides. So in the next slide, we can see the clear working of NAC when we combine NAC after the treatment by H2O2. So H2O2, when we treat it with H2O2, the cells, the microtubule cell structure is dislodged. So after treatment of NAC counterbalances the effect of H2O2 and its structure is restored. Here the comparison of the previous images in the control and in the cells treated with NAC, which is the B image. We can see that microtubules are in the classic structure of polymerized state. But when we treat it with H2O2, the spaces we can see that the structure is lost and they are depolymerized. And when treated with NAC plus H2O2, the structure is restored again. So coming to the conclusion, as we know that microtubules disassembly is a very important part of pathogens of FECD. So we induced oxidative stress and it showed the disassembly of the microtubules. And when NAC, which is an antioxidant, was added along with oxidant H2O2, it restored the microtubule structure. So future work contains to optimize the concentration of these oxidants like H2O2 and NAC, and we also need to focus on a P38 MAP kinase, which is a signaling pathway through which FECD pathogenesis to the next stage, and we need to optimize the concentration of its inhibitor for in vitro studies using MTT assay.
And the further thing is we need to investigate the functional effects of oxidative stress on microtubules, PMR, TJs in PCs with and without the exposure of antioxidants and p38 MAP kinase inhibitor. And we need to properly investigate p38 MAP kinase signaling pathway, which is a very important pathway that induces destabilization of microtubules, which leads to barrier integrity dysfunction and ultimately plays a very vital role in the pathogenesis of FECD. We would like to thank Bioinvent Lab, SIT and Dr. Sanjay Srinivas for their contribution in this project, and we would also like to thank NIT for providing us a platform to showcase our work. Thank you.
Thank you.
Any questions?
So what is the concentration of the peroxide that you have used?
Can you please say it again? I can't hear it. The concentration.
Yeah, what is the concentration of H2O2 that you have used?
Just 50 we have just 50 micromoles up to for 1 hour. We have kept it for 1 hour, the cells.
Okay. So can you explain this NAC plus H2O2, the dynamics in relation to polymerization? Like with and without NAC, the last, the balance
Yes.
C and D.
So when we add H2O2 alone into the microtubule, we can see that the microtubule is disassembled. H2O2 is an oxidative stress inducing agent, so it disrupts the microtubule assembly, and when NAC is added, which is an antioxidant helps in restoring, that is, it scavenges the superoxide produced by H2O2, and the structure of the microtubules can be restored.
Okay, so one more question. So you said about polymerization and disassembly of these tubules. Can you give me some more information on H2O2 interaction with the microtubule? What aspect, how it actually, what kind of binding site it interacts, or how does it disassembles?
We need to study the downstream signaling more. We haven't gone into deep, but the basic pathway is this: that when microtubules are subjected to, here in this paper they use nocardazole. We use H2O2 as an oxidant, so it disassembles the microtubules. So GS.
No, I want to know the chemistry behind the disassembly when you use this H2O2.
We need to yet learn the functional effects. How it, like, we need to know the pathways, signaling pathways. We have
Okay.
this contains in our future work.
Okay.
Any more questions? All right. Thank you, ma'am.
Thank you.
For the next presentation, I'd like to call Divyashree Dinesh. Is Divyashree Dinesh ready with the presentation?
Yes, ma'am.
All right, please go on.
Is my screen visible? Ma'am, is my screen visible? A very good afternoon to one and all present here. My name is Divyashree Dinesh and I'm from SIT. Today I'll be talking about my topic, nanoparticle-based intracellular delivery of microtubule stabilizers, overcome microtubule disassembly in corneal endothelium under hypothermia and cytokine stress. These are the contents of my discussion today. When we talk about the basic layers of cornea, the endothelial layer maintains corneal transparency by removing excess of water and has important role in the vision of a person. The endothelium layers cells are non-regenerative and hexagonal single layer. It maintains stromal detergescence. The average cell density according to the age group is given, and it can be seen that it decreases with increasing age. Moving on, there are many corneal disorders, and all of them warrant only corneal transplantation as there is no pharmacological treatment. There is also a high
demand for corneas, as the global supply of donor cornea is only 1 for 70 needed. And in a country like India, where there is a need of about 2 lakh corneas annually for transplantation, only 45,000 of them are donated, out of which 30% of them fail due to acute immune rejection or surgical trauma. The donated corneas are stored in hypothermic conditions, resulting in disruption of cell integrity as well as barrier dysfunction. In a normal cornea, there will be equal partition of fluid pump leak mechanism, whereas in a cornea stored in hypothermic condition, it is subjected to hypothermia and cytokine stress, which is hypothermia is during storage and cytokine stress is after transplantation. Both of this which leads to the microtubule disassembly, disruption of the PAMR, which is a periactinomyosin junctional ring, and eventually the breakdown of tight junctions. This causes an increase in the fluid leak, which results in the swelling of cornea and also causes decrease in the stromal transparency. We therefore have an unmet need. We have found that the failure of corneal transplantation is due to hypothermic stress and also during the storage of cornea and cytokine stress after transplantation. Therefore, we hypothesize a novel method of introducing microtubule stabilizing drugs by means of nanoparticles, which will help to prevent barrier disintegrity of corneal endothelial cells and help maintain stromal detergescence. The specific objectives are the preparation and characterization of drug-loaded nanoparticles, to conduct in vitro release studies of the drug-loaded nanoparticles at 37 degrees Celsius and 4 degrees Celsius to optimize the drug-loaded nanoparticles for rapid cellular uptake and also determine their efficiency in preventing the loss of barrier function of donor's corneal endothelium. Now moving on to the isolation of cornea, the porcine eyes were bought from the slaughterhouse and the cells were isolated by explant culture method. The corneal endothelial cells are then transferred and grown in a carbon dioxide incubator. This is the protocol for preparation of the paclitaxel loaded nanoparticles, which was prepared by the nanoprecipitation method, where the mass transfer takes place between two immiscible phases, where when the organic meets the aqueous layer, and it causes nucleation, which hence forms the nanoparticles. Here are the results obtained. The size of the particles were found to be around 95 nanometer with very good size distribution. The zeta potential of the nanoparticles were negative. Hence, we coated them with positively charged PLL, that is poly-L-lysine, so that the negatively charged cell membrane can uptake the positively charged particles, and the positive charge resulted to up to 25 millivolts. The TEM image of the paclitaxel loaded nanoparticles show that the particles are spherical in shape. The size of epithelium B nanoparticles are also found to be around 100 nanometer with good size distribution. The zeta potential of the nanoparticles was negative at minus 18 millivolts, but then after coating with PLL, it changed to positive 25 millivolts. The TEM image of epithelium B also shows the sphericity of the particles. Now moving on to the drug release kinetics of the PNPs, that is paclitaxel loaded nanoparticles at 37 degrees and 4 degrees Celsius at different intervals of time, we have found out that the encapsulation efficiency of paclitaxel in PNPs was about 91%. The 37 release studies followed first order kinetics, and the 4 degrees Celsius release studies followed 0th order. In both 37 and 4 degrees Celsius, we can see that there is an initial burst release of the drug, which is caused due to the diffusion of the drug from the nanoparticles into the medium. The drug release was more at 4 degrees Celsius rather than at 37 degrees Celsius. This is because the drug is bounded to the nanoparticles by physical absorption, which is mainly by hydrophobic interaction, and this interaction denatures at lower temperatures. Hence, the drug release is more at 4 degrees Celsius rather than 37 degrees Celsius. The same kinetics was observed for epithelium B. The encapsulation efficiency of ENPs were found to be 88%, and here also the drug release was more at 4 degrees Celsius. Once the release kinetics of drug-loaded nanoparticles was understood, the nanoparticles were internalized into corneal endothelial cells. These cells were grown till confluency and were incubated with the drug-loaded nanoparticles for different intervals of time, and then the cells were fixed with PFA and stained with DII and DAPI, and the cover slips were then mounted onto a glass slide and observed under confocal microscope. The process image here shows the results of internalization of drug-loaded nanoparticles. Here we see that the nanoparticles, which are shown by the green dots, were internalized into the cells after 6-hour incubation. The red color was stained by DII, which stains the cell membrane. The blue color was stained by DAPI, which stains the nucleus. And moving on, once we confirm that the cells were able to take up the nanoparticles, we use the drug-loaded nanoparticles for microtubule stabilization under TNF-alpha stress. Now, TNF-alpha stress comes then once after the transplantation is completed, the body, the allograft rejection takes place, and during that period of time, huge amounts of TNF-alpha is generated in the anterior chamber. In this also, the protocol is that cells were grown on the cover slips and incubated with the drug-loaded nanoparticles for 24 hours at 37 degrees Celsius, after which the uninternalized nanoparticles were removed and the drug release was continued for the next 24 hours. The TNF-alpha treatment was done at 37 degrees Celsius for 20 hours, and then the cells were fixed with 4% PFA, and the staining with tubulin and DAPI was done and observed under a confocal microscope. The result here is follows. In the control, the cells are not treated with drug-loaded nanoparticles. The control image shows that the microtubules exist as a fibrillary extension form around the nucleus to the cell periphery. The second image shows the loss of microtubule staining in the cellular periphery, which indicates the microtubule depolymerization in response to TNF-alpha treatment. The next image shows how the drugs stabilize the microtubules by polymerization. The image C and D only shows the drug, whereas the E and F shows the drug-loaded nanoparticles. And we can see that in E and F, the sustained drug release proved to be more effective than that of just the drug. The same studies were conducted to understand the effect of hypothermia on microtubules and how the drug-loaded nanoparticles can stabilize the effect. The results of the studies of hypothermia are as follows, where in the control, the untreated porcine corneal endothelial cells, the microtubules are seen as fibrillary structures, and whereas in figure B, which is shown under the effect of hypothermia, the disassembly of microtubules were seen in highly dispersed alpha-tubulin staining. Figure C and D shows the effect of the drugs, paclitaxel and epitheliol, and how it helps to prevent the microtubule disassembly, and figure E and F shows the prolonged drug release from the drug-loaded nanoparticles, and it can also be seen that it helps to prevent the microtubule disassembly. These are just the preliminary data, and we are looking forward to conduct this data for a longer period of time, and this is just to show that the trend has been established, and we have to look for prolonged periods of time. Our conclusions say that the average size of the drug-loaded nanoparticles were about 100 nanometer with a positive charge. The drug entrapment efficiency varied from 88 to 91%, and the drug release kinetics at 37 degrees Celsius followed first order, and for 4 degrees is followed 0th order kinetics. Internalization of drug-loaded nanoparticles in corneal endothelial cells was successful due to the positive charge of the nanoparticles. Due to intracellular drug release, the microtubules of porcine corneal endothelial cells were stabilized by PLL-coated drug-loaded nanoparticles. Our future work include immunocytochemistry studies on barrier integrity under hypothermic stress, and also the internalization of the drug-loaded nanoparticles under hypothermic stress and stabilization of microtubule disassembly. I would like to acknowledge Bioengineering Lab and SERB and ARVO for their grants, and also Dr. Sanjay Srinivas for their contributions. I would lastly like to thank NIT for giving me the opportunity to present here. Thank you.
Any questions?
Yeah. So, what is the role of the drug in the nanoparticle?
The drug is bound to the nanoparticle, and the nanoparticle will be added to the cells, corneal endothelial cells, so that the particle, once after transplantation, it will release the drug. It will at a sustained period, like I showed in the.
No. What is the role of drug?
The drug is a microtubule stabilizing drug.
Okay. My second question is, what happens to the nanoparticle?
The nanoparticle degrades after a period of 20 days.
Okay. So, what is the degradation mechanism? Any details?
It is the particles are made of PLGA, so they are biodegradable by the body enzymes.
Okay, fine.
Thank you so much. Next, I would like to thank you. Next, I would like to call Amitesh Shrivastava. Is Amitesh Shrivastava around? Yeah, hello. Please go on. It's not. Yeah, it is.
to the presentation. My topic of presentation is biomechanical stress analysis with printed polymer implant with yellow and pink. The scheme of presentation. Biomechanics. Interaction with the human body, and when we talk about the body, we consider whole body.
Sir, your voice is too low. Can you raise your voice?
Hello.
Yes, sir. Can you raise your voice? Because we are very low to hear your voice.
Am I audible, sir? Now?
Yeah, now it is audible.
Okay. So when we talk about the body, we mean whole body or any part under consideration. Then coming to the lower extremities, it generally includes the whole portion between hip joint or pelvis joint to ankle. Then we know that the thickness of femur is around 4 mm to 8 mm, and it varies. The length varies from 260 mm to 293 mm. It depends on person to person. Then coming to the material and method, in order to construct a 3D model of femur bone, I have used a CT scan of a healthy volunteer subjected to screening with no pathological history and trauma. So after processing the DICOM files that I have imported from CT scan machine, I have constructed this 3D model with the help of materialized mimics and 3-matic software, and after many post-processing operations and different operations, this structure is obtained, and I have successfully imported it in ANSYS space claim. Now in order to perform the analysis, I have selected a mechanical workbench, and in which I have meshed it in a tetrahedron 10 mesh structure with 3 mm of size. Then the total number of nodes were 2,10,438, and elements are 1,46,581. Convergency test is also performed before meshing. Then coming to the mechanical property of PLA, these are the mechanical properties of PLA, and further these are the mechanical properties of peak element. Then I have entered the custom data of these two some common properties in ANSYS workbench, and also after that I have applied a boundary condition in which the upper portion that is connected to hip joint is fixed, and the lower portion or the condylidal region of femur, I have applied 1000 Newton of ground reaction force. So after that I have got total deformation that is in PLA, that is 130 mm, and in peak it is 36.5 mm approx. Then I have also performed the directional deformation around the z-axis, because the simulation part of this femur bone consider the standing position of a person, so the orientation of this bone is in z-axis. So here the deformation is 36 mm in PLA and 10 mm in peak. Then coming to the equivalent stress that is generated around the volume of this femur structure, we can see the stem portion or the middle portion of femur get maximum stress because of the low cross section area and the higher load distribution. Then coming to the comparison part, equivalent stress versus total deformation shows that under same stress condition, the total deformation is PLA is slightly more than peak element, Then coming to the second comparison, the equivalent stress versus the directional deformation. This also shows the same thing, that in directional deformation also we are getting higher deformation in PLA. Then coming to the fourth reaction to the fixed point, we are getting higher deformation in PLA and lower deformation in peak. Then coming to the directional deformation, same trend is obtained. Then in discussion part, the deformation of femur is most likely to occur the narrow middle section that I have shown through diagram of equivalent stress. The critical region on femur bone extend from femoral condyles to head following the lateral medial location of bone. Condyles may get deformed under excessive load, and femur is likely to deform in z-axis in lateral and medial position. Coming to the conclusion, analysis shows results of stress distribution behavior of femur implant when a person is standing in full extension position of knee and pelvis joint. This shows that due to self-weight and ground reaction, condyles section aims to deform more often. Further, from stress evaluation, narrow section in femur stress concentration is at stem, and we have also find out that PLA at the same equivalent stress strength to get higher deformation than peak in both total and directional deformation. Under force reaction also PLA suffers more deformation than peak element. Then coming to the application part, this study helps in constructing or building assistive device for mobility like prosthesis, orthosis, or exoskeleton. Also through imaging technology and utilizing it for constructing 3D model, we can design better knee and pelvis joint implant. Also, better design for support system for lower extremity under pain can be processed with recent study. Then these are the references. That's all. Hello.
Yeah. Any questions?
Yeah, so can you explain me the details about the software that you have used, whether it is a finite volume or finite element, and the material property and the material property?
Yes, sir. Sir, I have used finite element analysis here. Hello.
Yeah, which software have you used, and some details about the methods?
ANSYS version 19 I have used. Initially, I have constructed 3D model by using materialized mimics and 3matics. Then I have used space claim in order to import geometry. Then in order to perform analysis, I have used ANSYS 19.3.
Can you comment on the material property of the bone?
Sir, actually, bone is much lighter and have a higher strength than PLA and peak. Rather, because polymer implants are lighter in weight, but the trends are here for metallic implant, which are quite heavier and requires more rehab time in order to.
Okay, thank you. Yeah, thank you very much.
Thank you so much. We wrap up the oral presentation session here. For the evaluation of oral presentation, I'd like to thank Professor Balasubramanian sir and Dr. A. Ravikant sir for sharing their valuable time with us today. With this, we come to the end of the third session. We'll meet again for the fourth session to start from 2 p.m. soon after the lunch break. Thank you so much, everyone.